पोंडा सर तो यहाँ आए थे 1-2 बार।
हाँ हाँ,
3 बार आए।
मेरा साथ फर्स्ट मीटिंग हुआ बनी बिहार में।
अच्छा अच्छा,
हाँ, नाउ आई रिमेम्बर।
यस।
I know Professor Patra.
So with all your permissions, we'll start the program. A very good morning to you all. It's a real pleasure to be with you all this morning. On the behalf of Department of Life Science, NIT Rourkela, I am delighted and honored to welcome all the dignitaries, admirable, honorable, and our dynamic director, Sir Professor Animesh Vishwas, sir. Our eminent chief guest, Professor Amulya Panda, director of NII, and all other eminent speakers and participants from India and outside. The very purpose of this meeting is basically to inspire the young minds and achievers to think beyond what we think is possible and push our limits to achieve more. What kind of medical facility and healthcare systems we can expect to evolve in next 50 years? Maybe we have to think it now and do research accordingly. In this pandemic situation, we have learned a lot, and it has definitely triggered us to revolutionize our current ideas of research and be prepared for any major changes that might affect us in future. So there might be many more unseen challenges, but we need to update our current understanding and to remain prepared for it. So there are many researches going around the world, but when it comes to the translation into the real-time scenario, there are many challenges, and often times it fails. So we should be aware of that, what's new and what's doable, how knowledge from one field can be applied to other fields, making less mistakes, and it is truly transferable in the benefit of humankind. So when it comes to the real-time inventions, every step actually needs to be reinvented, re-evolved, whether it's diagnosis, drug discovery, drug delivery, drug efficacy, patient handling, monitoring, screening, follow-ups. And these advances in these fields can truly revolutionize our current health. So hence, it's a 2-day conference program, and we will be listening to talks from various fields. And hope these 2 days will be a really learning platform. Coming to the theme, drug delivery is a broad field of research, and it's basically developing various kinds of novel materials and carrier systems for effective therapeutic delivery of drugs. And a steady control is needed, and patient monitoring of the data, monitoring of the things are required in a lot of respects. So in this area, a lot of scientists, young researchers, students, they are all involved. So, I encourage all of the students and participants to go through the lectures and be patient with the speakers. Definitely, they are going to enlighten us with their ideas, innovative things, and there will be a scope for interaction with our participants. So, during the time of presentation and as well as after the presentation, after the presentation. So, and later on also there will be scope for our students to interact with all the speakers. We can take forward their mails and we can connect them for better interactions. I hope everybody will enjoy. They will definitely get to learn. So because our speakers they are coming from various backgrounds, both research and academia. So hope the participants will have a know-how how to really transform the research into a product or a real-time application in health sciences. So with this, I would like our HOD, Dr. Surajit Das, to just take on and say some few words and encourage our participants and address the gathering. So, Professor Surajit Das.
Thank you, Bismita.
Hello, Professor Surajit Das.
I'm being muted. I'm being muted frequently. Why? Okay. Am I audible?
Yes, I can hear you. I think
Thank you, Bismita, for briefing the overall theme of this conference. And at this outset, I would extend our warm greetings to the chief guest, Dr. Amulya Panda, sir, who is the director of NII, the distinguished speakers, and all other participants to this national conference, present and future of drug delivery approaches and molecular medicine, organized by the Department of Life Science, NIT Rourkela. So as the convener, Dr. Bismita Naik, she has already mentioned drug delivery approaches and molecular medicine is an emerging and upcoming area of research. This is realized more during this ongoing pandemic situation. Drugs have long been used to improve the health. However, the practice of the drug delivery has changed dramatically over the years, and more significant changes are expected or anticipated in the near future. Molecular medicine is a broad field and where physical and chemical, biological, in bioinformatics, and several techniques are used to describe molecular structure and mechanisms. It also identifies fundamental molecular and genetic errors of disease and develop molecular interventions to correct them and elucidate disease pathogenesis at the molecular or physiological level for disease diagnosis, theranostic, and various other applications. Hence, in this connection, getting an updated understanding of the field is of immense importance, and this conference is the need of the hour, where the industry and academia, both speakers, they will be delivering their talks and will be enlightening us with their ongoing research findings. So, on behalf of the department, I extend again my heartfelt gratitude to the chief guest, Dr. Amulya Panda, and who is the eminent researcher and who has also been recently listed among 2% of the world distinguished researchers. And I hope that the talk from him and also the talks from the different participants will enlighten us with, and there will be a great learning platform in this two-day conference. I congratulate the team, the convener Dr. Bismita Naik and the treasurer Dr. Muthuswamy Srinivasan for their effort to hold this conference broadly on the drug delivery aspect and molecular medicine. I thank our esteemed director, Professor Animas Biswas sir, for giving his consent and for giving his constant support and encouragement and motivation to do the quality research and teaching. I hope all will be enjoying the talks and learn from the speakers. And I again want, on behalf of the Department of Life Science, NIT Rourkela, once again welcome you all to this conference. Thank you.
Thank you, Dr. Surajit Das. I'm very delighted opening remarks from you. So with this, I request Professor Samir Kumar Patra sir to say a few words about us for the conference and overall. Professor Samir Patra sir.
Good morning, one and all. Respected Director Sir, Professor Animas Biswas, our beloved Director, Dr. Amulya Panda Sir, Director, and our Chief Guest, Director of NII, National Institute of Immunology, New Delhi, and all my dear colleagues, the convener, Bismita Naik, Madam. I would like to brief little bit about the department first. The department was started in 2008, and then in 2009, the academic program started. At that time, 3-4 faculties took the initiative to develop the department, and Bismita, Surajit Das, Sujit Bhutia, and me, we took the initiatives to build this department as it is now, and it is going as per our dream. Although you know all the dreams certainly not fulfilled in life, but part of it, and it is expedite nowadays with a strong support from our beloved director, sir, Professor Animas Biswas. Thank you, sir, for your strong support to this department. And then about this program, the present and future of drug delivery approaches and molecular medicine. It is really in the frontiers areas of bioscience researches. Drug delivery, as we have learned, it is an art and it should be emphasized, because we have seen here in the COVID era, without knowing the nature of the virus, all around the globe, the clinicians, the doctors, the physicians, and or even any kind of healthcare personalities, they were used to apply any type of drugs that are used to cure the disease of humans. So, you know, without knowing anything, the drug approaches to cure diseases were started, and still we know the molecular nature, the what methodology, but yet when a new disease comes, we are in blind. So you all can understand the importance of drug delivery mechanism and mechanism of action. So once again, I thank Bismita madam for taking the initiative of such a nice, topic. And it is not the only one, it is the third on its nature in the department. Initially, it was started by two conveners, Rohan Dhiman and Vidyanegi madam, and then for plant, Vinod Bihari Sahu, and the third one is now by Bismita madam. So it's
Sir, it
really,
is fourth one.
yes, the fourth one. And you know, for all these events, you need a dynamic HOD. And I appreciate Dr. Surajit Das for every kind of support and encouragement and initiative for doing such of activities to further the science from our NIT Rourkela campus. And it also in this way, it is an art to bring the glory for the institute as well. Coming to the conference program, there are schedule has already been sent. I don't want to tell much. And I once again, I thank you all. Enjoy the 2 days seminar conference, national conference. And I thank you again, all, and all my beloved colleagues, our beloved director, sir, and our chief guest, Dr. Amulya Panda, sir. Thank you all.
Thank you, sir. Thank you for such nice words and encouraging words. You are always there with us, I know. From the beginning of this institute, this department, you are with us and constantly encouraging us for better things. So with this, I would like to request our director, sir, Professor Animas Vishwas, sir, to kindly address the gathering with his few words of inspiration. So
thank you, madam. Thank you, madam. Good morning to all of you. I'm proud to say that life science department is one of the most productive in terms of research in our institute. So I really admire these departments, and I wish all the faculty members to continue such a good research activities in future, present and future also. With this, I start this. I welcome the chief guest, Dr. Amulya Panda, Director, National Institute of Immunology, New Delhi, and all the distinguished speakers for different parts of the country who have agreed to deliver their talks on this webinar on present and future of drug delivery approaches and molecular medicines. I came to know that more than 440 participants have registered for this meeting, and live telecast is also happening through YouTube streaming. I congratulate the convener, Professor Bismita Nayak, and the head of department, Professor Surajit Das, for organizing this webinar. I am happy to share with you that the Department of Life Science and NIT Rourkela is making steady progress in both academic and research area. This department is popular destinations for various students for masters and PhD courses. This conference aims at converging knowledge from both academia and industry so that audience will be getting updated information on present scenario of drug delivery or drug discovery. I wish the convener good luck for these meetings and hope all the participants of this conference will also be benefited from the same and everyone will have a fruitful deliberations. With this, thank you all and wish you all the success of this webinar. Thank you.
thank you, sir. Thank you so much. You are always there. You have encouraging me and others and everyone in the department, and that's why we are actually progressing. Thanks a lot for your encouraging words. With this, without much delay, I would request our chief guest, Dr. Amulya Panda, who is also the director of National Institute of Technology, National Institute of Immunology, New Delhi, to share few words and address the gathering. So I will request Professor Amulya Panda to take over.
Good morning to all, and good morning, Professor Biswas, uh, Professor Patra, Professor Das, and Bismita. Thank you very much for giving me an opportunity to talk to you all people and give a talk. In fact, let me tell you, when Bismita called me, it's difficult to say no to your student, all of you know. And I had no option but to come. And I was talking to Professor Biswas. I have been attached to NIT long back, 10 years back. In fact, NIT is the only engineering college in the country who has a certified animal house. That is the beauty of NIT. You are good in engineering. The biology has to discovered, discussed in a physiological context, and that needs an animal house. I must congratulate the director and the faculty. I know Dr. Patra and the Department of Biotechnology, how they have struggled last 8 years, whatever it is, but you have indeed a full-fledged animal house, and that is the most beautiful things that could happen. And me being associated with it, being a part of the NIT team when it was being, I thought it will be nice to come and give a talk. And coming to the topic what Bismita has taken, we know, considering our country is poor and we have a different challenge, discovering novel things are very, very difficult for us, to be very frank. It is very, very difficult to discover entirely new drug, entirely new modality. So the only alternative for us is, can you do green technology? Can you deliver? The same drug can be put in a delivery concept. So in the future, when as you know, we are not discovering any antibiotic more, very few proteins are coming up. So the best way is, can we develop new delivery mechanism to make it more patient plans so that things can reach the people. I think on that context, drug delivery is really an emerging area, and drug delivery along with tissue engineering, scaffold and other things which you people are doing both in Department of Biotechnology and School of Biological Science. I think Department of Life Science, I think it is a wonderful idea. I am happy. Maybe in my talk, I do a little bit of vaccine delivery. Maybe I'll talk, but this is highly appropriate because coming back, when I give a talk on this drug delivery, I talk less on drug, but more on delivery. And delivery in a context that our education system, our engineering education, our science education, everything needs to be delivered to the people for which we stand. In that context, this symposium show a great importance. I wish all the best. I am sure all the faculty of life science. Uh, I don't see Suman here. I last time I came, one of his PhD student Viva. I see Dr. Patra, I see Surajit, I see Kuntia, I see Dhiman. All the best to all of you and have a wonderful time. Thank you very much.
Amulya Kumar Panda. Uh, Thank you, sir. Thank you so much. And with this, I think we are already delayed with the program schedule for half an hour. So probably with much delay, we would like to go for your presentation. But before that, I would like to request Professor Srinivasan sir to give the vote of thanks. Dr. Srinivasan sir, are you here?
Yeah. Yes, ma'am. Yeah, sure. So good morning to all. Our Honorable Director, sir, Chief Guest, Dr. Amulya K. Panda, sir, Director of NAI, and the Chairperson and Head of the Life Science Department, Professor Surajit Das, and Professor S.K. Patra, sir, and Professor Bismita, ma'am. So the convener of this conference and all the invited guests, as well as our faculty colleagues and all the participants. So it's my privilege to have been asked to propose a vote of thanks for this occasion. So I, on the behalf of NIT, the entire organizing team, and on my own behalf, I extend a very hearty vote of thanks to all the speakers for sharing their insights and their opinion today. And further, we are grateful to our Honorable Director, sir, for addressing the gathering and for his gracious participation in this inaugural session, besides his busy schedule. Sir, we all are inspired by your great words. And I may like to express our sincere thanks to our chief guest, Dr. Amulya Panda, for giving an excellent briefing on the importance of the drug delivery and the molecular medicine. So, sir, we are immensely glad to have you to grace this occasion. I also wish to express my gratitude to the chairperson, Professor Surajit Das, for his opening remarks of this conference. And we also would like to acknowledge our gratitude to Professor S.K. Patra for his directions and encouragement for organizing this event. And now it goes to the Professor Bismita Nayak, who has given shape to this event and make this event happen today. And well, an event like this, it cannot happen overnight. So the preparations have started weeks ago. So we have been fortunate to have backed by a team of very motivated students and technical staffs of Life Science Department as well as a computer center for organizing this event. And once again, I want to state that we all are most grateful to all the speakers, those who are present and those who are going to present the lectures. So we thank you. We thank you for being with us this wonderful morning. It's been a great pleasure. So thank you all very much.
Thank you so much, Professor Srinivasan. Professor Srinivasan, he has joined the latest joinee among the latest joinee, but he has been so dynamic to get involved in all aspects of Life Science Department. And the whole, as he said, definitely I'm very fortunate that I am in a department where it's more like a family and everyone supports each other. Together we go. So with this, I again thank you, Professor Srinivasan, for presenting his vote of thanks. Now I would like to request Professor Amulya Panda to present his talk, which is our keynote lecture for this session, morning session.
Okay.
So over to Professor Panda.
Okay. Let me share my screen.
Yeah, Bismita.
Yes, sir.
Introduction.
Sir, just hold on for a second, sir. Sorry, sir. Professor Amulya Panda, before he starts, I would like to request Professor Samit Kumar Patra to introduce Professor Amulya Panda to the audience, to the gathering.
Yeah, Bismita, let me load mine. Yes.
Yeah, while
sir, it is visible, sir. You can.
While Dr. Panda is loading his, slides, I would like to say a few words because he is in our heart, son of the soil from Odisha, and as a student, he was bright. I know he is graduated from IIT Madras in bioengineering, chemical engineering, and then he did his PhD from IIT Delhi, and working more than 30 years in the NII, that is National Institute of Immunology, and currently the director of this same institute. So this says enough about his charisma, his loving attitude to our department, all the faculty colleagues that you have known by interactions and all these. But yet I would like to mention some of his achievements because we never go rapidly or intentionally to see who is doing what. But here when he is with us, we have the opportunity to glorify and mention some of his achievements. That is, first of all, I would like to say that he has published more than 145 research and book chapter articles, and those which are very highly cited and they have scientific impacts. And then he presented in many conferences and others. He has 35, as much as 35 patents. And trademark products are 3, national awards 4, invited lectures he has given numerously. He has guided as much as 15 PhD students up to date. And he is still in his high time and producing very good science. And notably about his some of the honors like Samanta Chandra Shekhar Award from Orissa Vigyan Academy in the year 2000. He got it. And then from Department of Biotechnology, Young Asian Biotechnologist Prize from the Society of Biotechnology, Japan. Then Tata Innovation Fellowship in 2010 from the Department of Biotechnology, Government of India. Then G.B. Manjarekar Award of AMI at AMI 2010 conference at BIT Misra. He was the president of Association of Microbiologists of India, AMI. You see, he's a chemical engineer. And by his research caliber, he became the president of AMI. And Dr. Panda has the credentials of generating highest number of IPRs while working at National Institute of Immunology. It is a huge. We applaud, and I request you to start your lecture, sir, Dr. Amulya Panda. We are proud of you.
Okay, thank Bismita, I am audible?
Yes, sir. We are very much audible.
I'll continue.
Yes, sir.
So you could see my
Thank you,
screen, I can
Professor
move.
Patra. Yes, sir. Yes, sir.
Okay, go ahead. I have 40 minutes. So wonderful. What I'll do is I'll tell the title is Translation of Innovation in Development and Delivery of Biologicals. So what I'll do is, I think all of you, don't take it other way, all of you talk about innovation and translation nowadays. But I think it is important to understand what is this innovation means and what is this translation means. I may be looking very novice, but I like to put you my way of understanding what is innovation and translation. And this is particularly for the students, not for the faculty. And while talking what is innovation and translation, I'll give you two examples how innovation can be applied for making a recombinant protein and a innovative vaccines. So bear with me. Initially, most of you are professor, but the first session of my talk is only for students, because I believe anything you do, you must have a clear understanding of things, what you are talking. Everybody talk about innovation. Even 10 years back, everything was politics, everything was technical. Now everything is innovation. We must understand what is innovation and how innovation is different than a so-called jugaad. Somebody is making lassi using a washing machine. You may call that is innovation, but I'll tell you that is simply the logical way of extending the process of centrifugation. That's not innovation. So we'll go by that. Let's see what is innovation. These two lines you must have studied. Columbus discovered America, Galileo invented telescope, Edison innovated. So the difference between innovation and invention is, invention, you put money to do new things. That is invention. Innovation, I believe in a lighter term, is reverse of invention. In innovation, you use your science to create wealth. Invention is you put wealth to create knowledge. Innovation is you use your knowledge to create wealth. That's why Edison innovated. Of course, there are different ways of understanding, but you need to understand and contemplate very deeply to understand innovation. It's about a novel thing which has utility to the public use. With that, otherwise it is not innovation. And as I tell you, the most easiest way to remember or difference between invention and innovation is you put money to invent something. In innovation, you generate money. That's what Edison innovated electricity, because he was doing all his science, but what ultimately it gives a societal benefits. Let's go. What is translation? I think you are a school of life science. You know this nucleic acid to protein is translation. Let me go a little fundamental. When you go to nucleic acid to protein, two things happens. There is a physical change and there is a chemical change. Chemical change is nucleic acid from purine, pyrimidine goes to amino acid. I don't say nucleic acid doesn't have conformation. They have conformation, secondary structure of DNA, RNA. But you all know protein has conformation. That means when nucleic acid go to protein, you see a visibility change because proteins are conformation. And the chemical change, because this is nucleic acid, this is amino acid. If you look back and analyze what is a chemical change, in the school we have studied, chemical change is a permanent change. It's not a temporary change. It's not a hydrogen bond. It's a covalent bond. So chemical change is a permanent change, and physical change is a visible change. So to translate what actually is a translation in two terms, when you see permanent visible change in society, that is called translation. Making a clone somewhere, isolating a protein, or developing a molecule is not translation. It can be only called translation when the discovery or the invention, whatever you make, it really has an impact in society in terms of improving the quality of life or giving economic measure. Then only it is translation. Let's see. I call it as a rule of translation. See, everything in doing science not need to be translation. There should be always blue sky science, like Niels Bohr or Rutherford. They just wanted to understand how the atomic structure. But if you want science to be really useful, something like Louis Pasteur, you need develop something and something useful. So the first thing is it useful and needed? That is what is first thing. Is the science reproducible? Like I work in an immunology institute. You give me a protein, I'll immunize in 4 different ways, and I'll get 4 type of immune response. I'll get energy, I'll get immune response, I'll get tolerance, but that cannot be used as a technology. Then has the proof of principle has been validated in an appropriate model. See, when you do a biological science, many of you are life science, you need to cure the cancer in physiological milieu, not in cell culture. And please remember, mice is a mice, human is a human. Mice is not a small human being with a tail. Whatever you do in mice doesn't work in monkey. Whatever you work in monkey doesn't work in human. And even in a disease scenario, why you are struggling for coronavirus, even why you are struggling for HIV, why you are struggling for even developing disease model, drug and vaccines for many infectious diseases, because you don't understand the disease in a real model. What happens in a cell culture, what happens in the animal model is entirely different than what happens in a physiological study. Is it scalable? I think anything science you do, anything technology, if you do want to see that it is translation, it should be scalable fast. I'm sure all of you must have studied how to make oxygen in school. You take potassium chloride, add manganese dioxide, you heat it in an inclined tube, you get oxygen. Have you ever thought anybody, any industry making oxygen like that? No, that is not scalable. So what we have studied and what we realize is entirely different. Oxygen is purified by liquefying gas. Nobody, no industry make oxygen by heating potassium chloride and manganese dioxide. And finally, is it competitive? Remember, some 30 years back, when I was a student, I was punching card. Then came the 286 computer. Then come the floppy disks. Then come the USB. Now we have all cloud computing. So technology has to be improving, and technology has to be with the requirement. So that's why I say, when you want to do a translation, you take biology as the core subject. It's like a input-output reaction in a chemical reaction. You have input and you get the output like a chemical reaction. Then only you can think of achieving a translation. And when I talk about biology, I say biology has 4 types of molecules: protein, nucleic acid, carbohydrate, and lipids. We know a lot about protein, nucleic acid, maybe carbohydrates. Lipids we have just started to understand, but it is difficult to produce a drug using nucleic acid, carbohydrate, even though they're few. But proteins are ideal because you can use them to target and as well as drug itself. That's why I'll talk how you can do development and delivery using a protein drug or a carbohydrate. I wish I could talk about nucleic acid, because if you look at the current corona vaccine, most of them are nucleic acid products. Let's hope either one DNA or RNA comes up. Maybe we'll be able to tell what we are doing with nucleic acid. But I'll go about protein. Let's see how you can discover or deliver protein. That is important. This is all I want to say. If you take the whole living organisms in the world, all living organisms, plant, animal, insect, everything, virus, we have 20,000 protein function. And you know, all proteins are not druggable protein. There are muscle protein, there are other proteins. So out of these 20,000, 2,000 are druggable. Like you cannot use a collagen as a protein, but insulin is a drug, or maybe gamma interferon is a drug. Out of 2,000, till now, there are 200 protein in the market. So there are two options. Either you discover another 800 protein, which is already there. They may be in the sea, they may be in sky, in extremothermopile, or many places. Or if you cannot discover, that's a very costly thing that is time consuming, you use green technology. You make different formulation. Can you use delivery? Like insulin you take injection. Can you make a oral delivery of insulin? Or you take drugs. You take drugs every 3 days. Every day 3 times tetracycline. Can you take a single drug so that it mimics the 3 dose? So these are the things which coming. I'll give an example of a protein, recombinant insulin. Let's see. We know we are the diabetic capital of the world. Anybody who get type 1, you need 500 milligram of protein per year. I'm sure you get 20, 30 units. That's around 20 milligram per day, and 300 days around 500 milligram per year. Imagine Indian diabetic population is around 2030 will be 100 million. If 10% are diabetic, that is 10 million people into 500 milligram daily year. So we need 5 tons of insulin. You talk to a molecular biologist who express protein, he may be expressing in nanogram or microgram and milligram. We are talking of tons here. That is the level of scale we need. But that doesn't mean we cannot make. We'll be able to make it. Let's see how you can make this insulin. I'll give an example how to make this insulin a ton, maybe using technology. I'm sure somebody else is talking on biosimilar, but this is a way of telling how even with old thing you can do innovation. And we all know when you express E. coli, even the insulin is available in E. coli, inclusion body. And the moment you express it come as inclusion bodies are very bad, difficult to refold, and people, the moment you saw inclusion body expression, people leave it. They want soluble protein, or they want to do expression in different host, like you get soluble protein. Almost 20 years back, I thought, why not work on inclusion body protein? So one of my major research is to refold inclusion body protein, and this is how inclusion body proteins are purified. Normally, anybody who is in the lab, it's intracellular. You have the cell, you isolate cell, French press, you take the inclusion body, which are nothing but particles, you solubilize them in 8 molar urea. The moment you put 8 molar urea, what happens is, because this is highly denaturing substance, it unfolds the protein into random coil structure. You have a denatured protein which is soluble. Then you take the soluble protein and you refold it and purify. You see, if you do this, whatever you do, your efficiency is only 10 to 15%. Even insulin plant, which is produced by Eli Lilly from E. coli, recover 15%. Even in the lab, you may be expressing 100 milligram of protein as inclusion body. If you recover 10 milligram or 20 milligram of refolded bioactive protein, you must be doing a wonderful job. So here is a challenge. Can you recover more than 30%? Can you recover more than 40% or 50%? Anything you do. And many times people say when you do molecular biology or recombinant DNA technology, the molecular biology research is costly, this is costly, that is costly. But actually, if you see a insulin plant, because you refold at a very low dilution, actually urea and water are the most costliest thing in a recombinant plant. Because what you do is, am I audible, Bismita? Bismita?
Yes, sir, you are audible.
Okay, great. So what you do is, when you do a refolding at a very low dilution, low concentration, you use huge volume of vapor. Please remember, when you purify recombinant protein, you use ultra clear water, double distilled water, and that is contaminated with the DNA RNA. Even throwing that need treatment, so water is costly. So the real challenge here is, can you refold a protein in high concentration with less amount of water or with higher efficiency than 15%? I don't want to go in detail. You can check me with my papers. We develop a novel method of purifying protein. What we do is, see, protein is a beautiful thing, and people purify protein by using its intrinsic characteristics like its ionic charge, size, shape, hydrophobicity. We thought protein has another quality, it has density. Can you purify protein using its density? So what we do is, we isolate pure inclusion body from E. coli, because these are particles. To our surprise, when you isolate, we see that the inclusion body are nothing but only protein aggregates. So once you get the protein, pure protein inclusion body, but not having bioactivity, we solubilize them. We solubilize in a mild way and refold. In fact, this is the method I have been doing for last 20 years. You can check me in Google or maybe read some of my papers. We have been successful in recovering 50%. I showed you this is what. Left side is the E. coli expressing inclusion body protein. You could see dark points. This is the gel. And the right side are not E. coli. Right sides are the pure inclusion bodies. You could see how homogeneous these inclusion bodies are. Our idea is isolate this inclusion body with high purity, and we came to know that some of the protein inside the inclusion body has negative secondary structure. So we develop a solubilization method so that we don't disturb the existing structure. We don't use urea guanidine. In fact, at NI, we have developed and patented new method of high pH, beta-methoethanol, organic solvent, trifluoroethanol. So these are the different solvents we use. See, the idea is, inside the inclusion body aggregate, you have some rudimentary secondary structure of the protein. Don't disturb that. You just, if you know the amino acid sequence of the protein, you know what are the dominant force causing aggregation. You disturb them, make it soluble, refold, and you will get pure protein. In fact, with this model, I'm showing one example of human growth hormone. This is expressed in the left-hand side as the inclusion body. We purify the inclusion body and do all sort of chromatography, and you could see how pure protein we get. And if you see at the table from inclusion body, solubilization, refolding, ion exchange, gel filtration, we get around 40% recovery. In fact, I don't want to go in detail. Many protein, by using this method, we get 25 to 40% recovery. If you remember, a traditional method recover only 10%. So if you are making insulin, you want to make a ton of that. If you use the old method, you use 10% recovery, but here if you get 25% recovery, you are making more from the same clone rather than throwing it. So you will be doing a little bit of innovation to develop the new method of things. This is exactly what we did. And of course, I don't want to go in detail. These are my own books on different year I publish. The new book is coming on 2020 now on refolding of proteins. You can follow me, or maybe you can Google me. So the idea is, even if you develop this, can you purify insulin? That's what we did. We had an insulin clone. We isolate, we solubilize, we refold, and we try to see how it comes. See, it's not important what details the data I'm showing. It's what is important is the concept. The concept is, you purify inclusion body, you solubilize it without disturbing the secondary structure, and refold. I'll go ahead and show you some of my data. In fact, this is the protein. It may be the smallest protein, the oldest recombinant protein, but it is one of the most difficult protein, I tell you. It has H and B chain, it has 2 disulfide bond, 2 intrinsic bond, and actually in the body, it leaves, it stay as a hexamer. You can see in the right hand side, the left hand side is 1 A and B chain, how it looks like. You have a C domain, and this hexamer remain same. So what we did is, this is more or less like how H and B chain, but what we see that recently, in a couple of years back, actually how refolding takes place in the endoplasmic reticulum of a human cell was published. We exactly copy. What we did is, you could see in the right hand side, the insulin is synthesized from here. It comes as a B, C chain, it folds, make alpha helix, At the end, disulfides are formed. Once the disulfides are formed, the C chain is clipped and you get matured insulin. We exactly follow the in vivo method in vitro. I'll show you what we did. We express this, as I told you, I was showing the inclusion body. We get around huge amount of inclusion body aggregate. We exactly follow this. I'm not going to in detail, but we could purify the way exactly the pro-insulin C. But what you could show that we're expressing insulin as inclusion body in fermentation around 10 gram and 50% is the C chain. So if you cleave C chain, it is around 4 grams per liter of inclusion body. And if you remove when you start refolding, we are getting around 25 to 30%. So that means we are getting around 1 gram per liter, and that is very huge. In E. coli, 1 gram per liter in 1 day of fermentation. So if you are getting 1 gram per liter, you need 4 of 5,000 liter because it's almost a week batch. In 50 batches, you can have 1 ton of insulin. In fact, we are implementing this in industry. All I want to say that with an innovative idea, you still can make a protein insulin, which is things. And you may say insulin is a biosimilar, insulin is an old protein. There is no innovation in that. Let me tell you what is happening. So coming back, what we did in insulin is, it is needed. We develop a process which is reproducible. We have a proof of principle. Is it scalable, I showed, and it is competitive because we are getting more protein now. This is what called translation. And to tell you, coming back to insulin, you see, you can say this is a biosimilar product, but you can still do innovation. What you can do, please remember the below line. The same insulin molecule, 4 times Nobel Prize have been awarded for insulin. One, when Bantic and fellows discovered it, 1921, the first radioimmunoassay was used insulin. The first amino acid sequence was done using insulin gene, and the insulin gene was expressed. So 4 times Nobel Prize has been gone. If 4 times can be got, one more time somebody can get Nobel Prize if you do like this. Maybe a novel way of purification or a novel way of delivering. A couple of years back, Pfizer wanted to have a inhalable insulin. That didn't work. People are still working on oral delivery of insulin. That come to the contest where this symposium is organized, delivery of drug. If anybody could develop a delivery system for insulin, it will be wonderful, not for society, but worth scientific discovery. All I want to say that, what is the role of delivery, even if the molecule is old, even the insulin problem is old, even if the protein is old. With this, I go to vaccine delivery. That was a part of protein. I will come to vaccine, particularly because this is part of a delivery system. I'll tell you how delivery of vaccine can do. But before that, as usual, these are certain information maybe for the students, though, and to tell you very clearly, this is not 30 vaccines, this is 300 vaccines. There are 300 vaccines available both for veterinary and human use, but it is safe. It is one of the safest thing which is available for 4th century. Let me tell you, in a drug treatment or in a modality of infection, around early century, we developed sulfur drug. They got outdated. Then we discovered antibiotic. They become resistant. Then we become biologics. That's not working. We are making now conjugated antibody drug conjugates. Now cell therapy is coming. But vaccine is the only one modality of treatment which has not shown any resistance. Everything has shown resistance except vaccine. So that is the beauty of vaccine. For last 400 years, no resistance has been reported. You see the same TT vaccine we have been taking for last 100 years now. 1921, it was used along with alum, but now it's almost 100 years, not a single report of resistance. What my grandfather took, maybe my granddaughter will take the same vaccine. That is the beauty of vaccines. And when I tell about vaccine, I always give this example of smallpox. Look at this is vaccine titer in humans over a period of time. And look at the x-axis. It's not days, weeks, or month. It is years. Somebody has, this is in Faroe Island. Somebody has assayed the antibody titer of human beings for 60 years. And you could one single dose of smallpox vaccines maintain the antibody and protect the human beings from smallpox for 60 years. Think like that. Learn from smallpox or the live attenuated vaccine. Can we develop similar vaccine for TB, malaria, or maybe for COVID? That is what. When you say about vaccine, it do two things. It provides a neutralizing antibody titers, but more importantly, it gives memory antibody titers, because this vaccine is working because it produces immunological memory. Unless that, we don't call it as a vaccine. So to come back, vaccines are cost-effective treatment, but there are problem. We need repeated injection. If you have to take tetanus vaccine, you have to take 2. If you have to take hepatitis B, you have to take 3, 0, 1, and 3 months. If you get a dog bite, you take 5, or maybe previously you used to have 14. These are needed because when you immunize fast, you get tighter. But when you give the second booster, the immune system is activated, and that's how memory and other things come. So in those concepts, the concept of delivery is really, really important, because unless you give the second dose or third dose, there will be no permanent, memory titers or antibody titers. So the question we ask, this is part of delivery system I like to talk. The question we asked a couple of years back, can this problem of vaccine, repeated injection and giving more injection to the people in a single dose can be solved by using polymeric particle delivery system? So that is what. So what we are asking is, you have TT injection, you give 2 injection, 1 and 28 days. You have hepatitis B injection, you give 0, 1 and 3 months. Rather than give 3 injection, design a delivery system in which the vaccine is packed in a nanoparticle or a microparticle or a polymer particle or a liposome. You give a single injection and design the formulation, the delivery system in such a that it mimics the natural immunization schedule. For example, if you are giving it for TT, let it release everything in 0 and 28 days. If it is releasing, you are using for hepatitis B, let it release in 0, 1 and 6 months. So this can be monitored by taking care of the polymeric formulation, using their molecular weight. Higher molecular weight will grow, degrade slower, lower molecular weight degrade fast, many things. But what is important is this delivery system show a promise of developing single dose vaccine formulation. I'll not tell much, but I'll tell you, we have been working on this PLA, PLGA based vaccine delivery system. In fact, Bismita did her PhD on delivering rotavirus vaccine using PLGA particle. People are delivering vaccine, toxoid, recombinant protein, but she could deliver the virus, which is a bigger size than any molecules. However, these polymeric particles are FDA approved. They do multiple things. I don't want to go in detail. Again, you can check me in my Google. But we have been, just to tell you, these are the particles it looks like. And look at the size. It can be nanometer, micrometer, and macrometer. The left-hand side is nanometer. These are useful for delivering intracellularly. This micron size will be staying in between cell wall and membrane fusion. This macro size, like 90 micron, which are much, much bigger than the cell, will be as a depot system. In fact, you can play around with these polymer particles which you are working. You can play around with the surface, the molecular weight, the degradation profile, the size and shape, so that you can improve the interaction. You can deliver them intracellularly. You can deliver them to lysosome or to phagosome or to cell membrane. Many things can be done. And this gives a wonderful opportunity. And as I told you, we have been doing it many things. This is what is one of my major research in NIE for last 30 years, 25 years. So I have been working on DT, TT, hepatitis, and many things. We do multiple things. We do single dose vaccine, modulation of immune response, dendritic cell activation, many things. I don't think I'll be able to tell much. But one thing I tell you, as I told earlier, for developing any therapeutics, you have only 4 things: protein, carbohydrate, lipid, and DNA. The last example I gave a protein. I will give an example here, a carbohydrate as a drug and how to deliver it. So to tell you, we have done all these things with many details with the TT, hepatitis B, and we have filed 12 patents and publications. All this data, I'll just show you one data of TT. This is an antibody titers giving the vaccine delivery using microparticle with alum. Look, we gave a single injection, and this is compared with the 2 injection of MP alum, the square one. We leave the animal for 1 year, just like that, by giving a single injection. After 1 year, we boosted just to check memory response, and look what the memory response coming with polymer particle based delivery system, very high, indicating that when you use this polymeric particle based delivery system like PLA, PLGA, we improve the immunogenicity of the antigen. That means we can achieve same type of titer or therapeutic requirement using small dose. That is very important. So but the bottom line is, you use this nanoparticle or microparticle, you deliver them, they go to the cell, they do something different, and some new thing comes. But the real question is not making nanoparticle. Nowadays, everybody make nanoparticle, and nanoparticle enter cells, they'll deliver intercellular. But the real question is, what great thing it does? Can you do something different? Please remember, these delivery things, people think somebody discovered the drug, somebody formulated the drug, the delivery at the end of innovation. He or she will package them in nanoparticle, and then it will work or not work. What we are now suggesting and advocating is, delivery is a part of invention. Because when you discover a drug or a vaccine or a biological, there may be some inherent problem. It may be toxic, it may be degrading, it may not be stable, it may be not immunogenic. Can you use this delivery system as a part of to innovate something? In fact, these are the questions we ask, taking the clue that polymeric system deliver antigen and improve immunogenicity. Can it be a part of new vaccine development? Can you improve the immunogenicity of weak antigen? Can you do immunological memory? Can it improve the immunogenicity of a T independent antigen like carbohydrates? Many carbohydrates are not immunogenic at all. Can you do that? Can you modulate immune response? But these are the questions. See, developing tools and technique like drug delivery or vaccine delivery is one thing, but asking or using these tools and techniques to ask fundamental question in science is a beautiful thing, this delivery system. I'll show you one example how this delivery system can be used to ask fundamental question while doing the delivery job. So as you know, these vaccine delivery things improve the immunogenicity. So what I am saying is, asking polymeric system improve the immunogenicity of the antigen. So the question you ask, can it improve the immunogenicity of polysaccharide based vaccine like typhoid vaccine, meningococcal vaccine, or pneumococcal vaccine? These are polysaccharide. They are not immunogenic. They don't give immunological memory. That's why I tell why polysaccharide. They are weakly immunogenic. They are T independent. They don't give memory. They basically doesn't involve T cell. Mostly it is B cell response. Mostly it is IgM response. And there is no affinity maturation. It is only IgM, no IgG or other subtypes. So the question we are asking, if at all this vaccine delivery system is improving the immunogenicity, can it really improve immunogenicity of a carbohydrate? That will be a real path breaking. And that's exactly what we did to tell you what these carbohydrate vaccines are. If you see, the conjugate vaccines are always protein and carbohydrate conjugate, whether it is influenza or meningitis or salmonella or pneumonia, most of them are conjugated to TT, DT, because these conjugate vaccines improve the immunogenicity, because conjugate is a protein part. And the protein part, we all understand, protein part get present and processed through T cell and give T cell help, but carbohydrate doesn't do. So that's why all conjugate vaccine has both protein and carbohydrate part. But however, we want to ask a question, can you use this delivery system to improve the immunogenicity of carbohydrate without conjugating to a protein part? That will be a real game changer. So what we did, we thought, let's put this carbohydrate, put them in nanoparticle and deliver inside. Remember, this carbohydrate, carbohydrate is bigger, huge molecular weight. They don't enter antigen presenting cell. If they don't enter, the chances of presentation, processing, and getting TH1 or TH2 response is very, very weak. So we thought we can deliver this carbohydrate inside a polymer particle and ask the question, will it give protection? That's what exactly we did. What we did is, we asked the same question, can polymeric nanoparticle be an alternative to conjugate vaccine in terms of getting IgG and memory? Remember, if you immunize a polysaccharide, you don't get IgG, you don't get memory. So to do that proof of principle, we took pneumonia polysaccharide, PCP1. In fact, the pneumonia vaccine is 14 more. and we entrap them in polymeric particle. This is a tough carbohydrate. We struggle to put it back into the carbohydrate polymer particle. But once we made a particle, we thought, let's see whether alone particle can be substituted to a conjugate vaccine. And these are the pneumococcal vaccine. You can see 7-valent, 9-valent. These are different serotypes involved. 13-valent. These are 1, 2, 3, 4, 5, 6 are the serotype prevalent in India. What people do is take the polysaccharide, conjugate to CRM. This is a mutated version of diphtheria toxin, and that conjugate vaccine, whether it is 7-valent, 9-valent, is used. Our concept was, can you use the delivery system to discover new things? In other way, can you use polymeric delivery system to deliver the carbohydrate alone and raise antibody response, which will be comparable to conjugate, or will be neutralizing or giving antibody switching? So what we did is we made these nanoparticle polylactide, entrapping a carbohydrate, one serotype carbohydrate of Streptococcus pneumonia, and we immunize the animals and look for memory response. Look at this titer. This is PCP1 in microparticle, nanoparticle, and soluble. The primary, remember, carbohydrates are poorly immunogenic, so we don't get primary response. We leave the animal for 6 months, and after 6 months, when you boosted, look at the soluble carbohydrate, no memory, but you are getting memory in nano or microparticle. In other way, what we could show that if you deliver carbohydrate and immunize the carbohydrate entrapped nanoparticle, you will get memory antibody response. And these are all IgG. As I told you, we get IgM, but preferably we get IgG, indicating that when you use a nanoparticle-based delivery system, we achieve two things. We do isotype switching from IgM to IgG, and we get a memory antibody response. In fact, that was very, very exciting. In fact, we check the sera for option of phagocytic activity. That is the activity you take by taking the sera and putting them in macrophage and seeing how many bacteria these sera can neutralize. You could see cells with bacteria and PCB nanoparticle serum. It can completely neutralize, as shown in facts, indicating that what antibody we are raising with polymer particles is not only high titer, but it is neutralizing and has good binding capacity. In fact, we did a challenge experiment. We had a challenge facility here. We immunize the animals. Once the antibody titers is raised, we challenge them with lethal pneumonia, Streptococcus pneumonia. And you could see, look at the nanoparticle group, complete protection, indicating that when you deliver the carbohydrate in nanoparticle, not only it raises antibody titers, it gives IgG, and it gives memory antibody titers, which are neutralizing and protect the animal in challenge experiment. This is just like proof of pudding is in eating. This is the result showing that indeed you can deliver carbohydrate and you get neutralizing antibody titers. So to conclude, what you can do is entrapping a polysaccharide in polylactic particles induce serotype specific immunity from a single dose and get protection. We don't know exactly. We are studying whether it is intracellular delivery or it is activating germinal center. My students are studying. But to tell you how exactly do, we could find out these nanoparticles. You can entrap any dye, maybe coumarin or FITC, and check whether the nanoparticles are getting intracellular. You could find out there is higher uptake of nanoparticle in comparison to microparticle. That's why it may be a reason, because remember, these carbohydrates are antiphagocytic. They don't enter the antigen presenting cell. Probably using a nanoparticle-based delivery system, what you have done is we have allowed the carbohydrate to enter the cell, which otherwise would have difficult. So delivery is the key mechanism. And this is how, this is microparticle. You could see with a macrophage, it is staying in the surface. You could see nanoparticle in the lower layer. Nanoparticles are completely engulfed by the macrophage. There is change in cytoskeleton. So whenever you use a nanoparticle, many things happen. It enters the cell, it delivers intracellularly and do ATH1 kind of things, and maybe activate immune system. So all I want to say that while using delivery is important, I am showing here, using the delivery system, you can deliver carbohydrate, which was otherwise difficult. Not only you can deliver the carbohydrate, by delivering them intracellularly, you could raise antibody titers, which are high, which are neutralizing, and which have high avidity and activity. We don't know this delivery system, how the particle-based delivery system work on immune system. Depending on size of the particle, whether they are nanometer, micrometer, or very small, they can grow through a different way. They can go through phagocytosis, maybe carbon clathrin coated vesicles, or maybe pinocytes, many other way. So basically all I'm saying, using the same antigen can be delivered through different route using different size. It may have a depot effect. It may have, because the polymer particle or any nanoparticle have repetitive surface antigen, repetitive entrapment, it may mimic a virus. See, one of the beauty of virus is it has repeated antigen in the surface, something like coronavirus or any virus. If you look at the surface protein, the same antigenic moieties are repeated. So one of the requirement of this virus is having repeated antigen. The same way we are mimicking in the particle, and the other method is it can do cross presentation. Cross presentation means it can do both TH1 and TH2 presentation. It can bypass from phagosome to lysosome to cytoplasm. From cytoplasm, it can be processed through MHC class 1 way. So these are the different mechanism going on. And I want to tell you, apart from delivering these carbohydrate, what this nanoparticle being developed. See, as I told you, developing a formulation, delivering is something, but doing innovative things is the most important thing. What you can do innovative things with this nanoparticle is you can have intracellular infection, you can use mucosal vaccine, you can use vaccine against allergy. In an allergy, you need predominantly TH2, you want to shift it to TH1, you can use the nanoparticle, you can activate dendritic cell, you can activate the tumor T-independent antigen. Nowadays, these polymer particles are used as artificial antigen presenting cell. You see what an antigen presenting cell does. It activates T cells. So what people are doing is using this nanoparticle delivery system as a source of antigen presenting cell. They entrap different cytokine depending on what type of T cell you want. And as those cytokines are released, they activate T cell to a particular lineage. And these are many things going on. I'd like to end here. With that, let me thank you. I have used number of students who have been working and many collaborators. Most of them are from pharmacy. You could see Bismita's name somewhere because she worked almost 10-15 years back. New students are coming. With this, I'd like to end. But before I end, let me give you a message. Look at this figure. This mother has the food, but the kid is still hungry. Look at the kid. He's still looking for the mother to deliver the food. All I want to say, you may have a wonderful technology, as I told you in the definition of translation. Nothing matters if it doesn't reach to the people where it is needed. So the mother has the food, but the kid is still hungry because it has not been delivered. So delivery is the key. Thank you very much. You can ask me a question now, if anybody wants.
Yes. Dr. Panda sir, it's a nice lecture. And a lot of information for the students as well as for the scientists. And I want to deliver a message to the students. You see, being a chemical engineer, he is now immunologist, biochemist, biologist, everything. So it is your own wish and motivation, the way you want to go. So definitely, Dr. Amulya Panda is a motivation for the young generation. And I thank you with a much of applause, sir, for giving your time, valuable time for discussion and remaining with us. Thank you, sir. And the lecture is open for discussion. Bismita madam, you please take care.
Yes, sir. Thank you, sir. Thank you, Professor Patra. Thank you, Professor Amulya Panda, sir, for delivering such a nice talk. So with here, I would request Panda sir to just take off the slides from sharing so that we can see him fully. And I think the session is yes, sir. So I think the session is now open for discussion.
Can you see me now?
Yes, sir. I can see you. I think everyone can see you. I think we can take on questions. I have some few questions from YouTube, but here I would like to open if someone is interested in any questions here. So before we see anything, just a question from YouTube channel.
Can I ask one question?
Yes, ma'am. Sure. Please.
Good morning, sir. Good morning, Panda sir. It was a wonderful talk and like flooded with a lot of information. This is Vidya. I'm
Yeah,
asking the
yeah. Tell
question, sir.
me.
Sir,
Sir,
my
Vidya
question.
ma'am is
Okay,
from
go
our
ahead.
department. Yes, she's our department.
Yeah, I
faculty.
know, I know.
Yeah.
Sir, my question is when we are doing this delivery of this drug, when we make it like target specific, for example, if you want to target some cancer cell or some specifically infectious cell, which has been infected cell, not infectious cell, how easy is that task? Like making these nanoparticle or any delivery system to be specific for a cell?
Okay, if you ask me in true sense of target, if you understand want to target is target, like I target you, you are the target, that's all. But that's what Paul Ehrlich told 100 years back. We have not achieved that because when it goes to a cellular milieu, um, many things there. But what people are trying now, suppose you have a cancer or you are a particular thing, you can use a marker of that particular cancer or a receptor, and you can code the particles so that preferentially it goes there. Till now, nothing like 200% targeting, but preferentially this particle can go if you have in the surface of a particle a particular receptor for a particular cancer, like for a particular cancer, if certain receptors are expressed in cancer cell, the analog of that can be coated in the particle so that the particle go. But I tell you, still the real target in true sense is a dream for most of the delivery people. The size will go, there will be some hydrophobicity, some functional group you can attach. They will do, but they will also attach to other things, because remember, what you do in a biological system is entirely different than physiological system.
Right, sir.
But you can always try with uh, uh, um, different moiety in the particle surface.
Okay, right, sir. Thank you. Thank you, sir.
Good morning, sir. Another question.
Yeah, go ahead.
Okay.
Yes.
Sir, it was a wonderful lecture, and all the information was a lot of information we got. But I want to ask some questions regarding the toxicity of these nanoparticles, because actually we are working in our lab related to the nanoparticle toxicity. And this PLGA and PLA nanoparticles, they have shown to induce the ROS production and many apoptosis and all the other mechanisms which can potentially lead to cell death. We are doing it specifically in Drosophila right now, and we have a paper on the way. But the point is it safe that we can deliver this polymer, this drugs in polymer, because it has significant amount of toxicity as well. So, what is your comment on this?
Okay. There are, I don't know whether you know, there are 6 products already available using PLA PLG, and PLA PLG is nothing. But these are the same polymer where sutures are made. So it has more than 60 years of safety in human beings. Every time you go to the doctor, if he stitch something, that dhaga is nothing but PLA PLG. No doubt, when you have nanoparticle, if you do a cellular system, they will enter. I wish I could have showed you. We have done a lot of things on toxicity. These nanoparticles do enter the cell. They do all ROS production. In fact, I have seen the macrophage size increases. But then they get exocytes also. The cell has the tremendous capacity of absorbing this shock. Unless it is killing and doing, it's like a poison, it's not. Because this is a biodegradable polymer, this is a safe polymer. Because of the nanoparticles, there is disturbance. But they get exocytes from the cell also. So indeed, it is toxic to within the range in which you want to do. It's like you inhale the dirty air. Around 100 ppm is okay. But if you start inhaling from a chimney, you will be in trouble. But I don't think these nanoparticle, whoever is using drug delivery, use that amount. Maximum use 1 or 2 milligram of things because you are using very hyperactive drugs, and at that level in a body situation, it won't be that much toxic. And otherwise, body has the system to get rid of the toxicity also.
Thank you, sir. Thank you very much.
Sir, we have another question from YouTube channel. So, Prabhat Kumar Swai is asking, what is the importance of disulfide bond in refolding technology?
Oh, it's very, very important. People still don't understand how the disulfide mismatch, particularly with insulin. If you see the A and B chains are so close, any mismatch will make the conformation different. As you know, protein works only with proper conformation. So the importance of disulfides are very, very important, because if it doesn't make intradisulfide, the conformation will be different. If it make interdisulfide, you'll get dimer trimer. So any disulfide mismatch will lead to unwanted protein having unwanted topological things. See, one of the major problem why while refolding protein we lose lot, because this disulfide bond mismatch disulfide bond formation leads to aggregates, both aggregates which are visible, aggregates which are soluble aggregates also. So that reduces the efficiency. In fact, anybody, I didn't show much detail. We do a Elman method to know how much of oxidized reduced SS group is there before refolding, after refolding, just to get sure that maximum of them should have been refolded. And again, the disulfide may be important, but then you have to realize, if the disulfide is playing a major role in the alpha helical and beta sheet structure of the protein you are talking. Like insulin, it is important. If you take growth hormone, like I work in growth hormone, you remove these two disulfide, whether they are oxidized, reduced, nothing happens to biological, because the alpha helix structure is thermodynamically so stable, it doesn't allow the disulfide. But it is important in many, many protein where the conformation is controlled.
So if we can take some one or two questions more, Dr. Anand
Sure.
Basu from Department of Marine Biotechnology, BDU, he is asking how the fusion tax helps the recombinant protein overexpression and purification. The fusion tax, basically.
Sir, you is muted. Your mic is muted, sir.
Okay, you can Google me or check my public.
Sir, now I think you muted yourself. Just check once again.
No. Is it now I'm audible?
Now audible.
Yes,
Now audible, sir.
yes, sir.
Okay. So basically, if you see recently we have published, ideally you should not use a tag, because tag protein are not approved for any human use. Any histidine tag, Mali, or any tag, they are easy for purification, but they are not allowed by regulatory body, because you see, you inject the tag protein. Suppose you have a malaria vaccine made up of histidine tag, you inject. The body will make antibody against the protein, and the body will also make antibody against the histidine. So your histidine antibody is circulating. Suppose tomorrow another fellow develop a pneumococcal vaccine with tag. The moment he immunized, already histidine tag antibody or tag antibodies are circulating. They will neutralize your vaccine. So tag is not approved for any human use. So it's better not to use tag. Please remember, much before the recombinant thing came, people were still purifying protein, isn't it? It's such a beautiful molecule. You have the size, shape, density, metal binding characteristics you can purify. So tags does influence the immunological part, the structural part, many things, because you have extra protein, you don't know what that extra protein will do to your body. Okay. I am not able to hear you.
Suman sir, are you there? You want to interact with sir, or should we take another question?
Suman is there? I don't see him.
Sir, he is there, but I think he's muted. So another question.
Yeah, sir. Good morning, sir. I had only one question. Sir, I'm fine. How are you, sir?
I'm fine.
Sir,
Your camera is on?
No, sir. Let me switch on the camera.
Yeah, that will be nice to see you in blood and face.
Thank you, sir. Sir, what if we can deliver carbohydrate polymer without conjugating it with protein? or say any
Yes.
proteinaceous toxoid. So how will it help with the immune response? I mean, immunogenicity.
No. See, what we are doing now here is generally all carbohydrate vaccines are made up of carbohydrate and a protein, because the protein gives T cell health, right? That's why carbohydrate, if you immunize, you get antibody, you don't get memory and you don't get IgG. What we have observed, when you entrap this carbohydrate alone and put them in particle, the antibody titers you get are IgG and neutralizing and memory. We are still studying. Recently, one of our students has done this particle go to the germinal center, activate T follicular helper cell and other thing. The mechanism we don't know, but we certainly are sure that because of this nanoparticle based delivery system, the carbohydrate which are antiphagocytic doesn't enter antigen presenting cell, is able to enter because the nanoparticle is entering the cell. And while it is entering, it is delivering. What it does inside, we still don't know. But we get good neutralizing antibody titer. Is that clear your doubt?
So as you said, no, sir. So as you said during lecture that if we can deliver it without conjugating with the proteinaceous toxoid, it will have better. We will have better, immunogenicity. So how will it help with immunogenicity if we can deliver without proteinaceous toxoid attached to it?
No. If you attach any toxoid or any protein carrier to the carbohydrate, the conjugate itself will be immunogenic. That's what the conjugate vaccines are available. What we are doing is, because that is costly, we are 1 minute. We are delivering without conjugating, and we are getting the titer.
Okay. That's the only reason we need to. We are looking for the
the real molecular mechanism. The real molecular mechanism, we don't know.
Okay, sir. Yeah, that's all. So thank you for the inspiring talk.
Sir, we have a last question and then we'll move forward with the other presentation.
You better give me.
Yes.
Have
So a quick question actually from Ashwin Kumar. He is asking, like, if we change some amino acids, short acting and long acting amino acids in the sequence of insulin, is it going to impact in the purification process, insulin purification process?
No, that has people have tried, because if you I don't know whether you know it or not, the real insulin is the H and B chain, but there is glargine, there is aspartic, so 1 or 2 amino acid has been put extra. They change the charge, they help in delivery and slow release. If you see, there are long acting and slow acting insulin. That has been achieved by putting asparagine and aspartic acid, because it increases the charge. But that has not helped the purification per se. That because the pH became, PI of the insulin became very close to the blood, so it microaggregates, it slowly releases. But a purification, no. Anything you add extra, it is undesirable, it is not allowable by regulatory body, because you have to make the real protein as such. You cannot make extra protein and remove it. But it will not help in much, because at the end of it, you have to remove that protein also. It may not help.
Sir, we have a question, and it's a question from my side also. While we are delivering, how the HLAs, they influence the overall delivery of nanoparticles. Let's say we are formulating many, how allergenetically or immunologically they will be, how we can control that HLA factors. Like, is it going to work in all?
HLA may not be a major factor, because what you are using a vehicle which is not immunogenic, like PLA, PLG or any other thing. But if the protein or a drug which you are delivering, that affects or that has something to do with your genomics, obviously it may improve or reduce the effect. It all depends on the drug and the content.
So similar question Mrityunjay is actually asking, like oral delivery of insulin like proteins in camel milk has significantly improved the condition of hypoglycemia. So can we see any potential advances using such kind of insulin proteins directly in humans for future use like that? So is there any genetic?
You have to understand. No, you see, understand. Till now, there is no oral insulin because the insulin is unstable at low pH. The moment you eat at the stomach pH, 0.2, 2 pH or 1.5 pH, it degrades and it loses its immunogenicity or things like that. What this camel milk may be doing, I don't know about this, but what this camel milk may be doing, see, camel milk or any milk has a lot of protein, all this casein and other things, and fat globule. So this protein may be getting protected. The insulin may be getting protected from the stomach digestion because of the enhanced presence of milk protein. In fact, people and once the milk protein along with the insulin passes through the stomach, once it goes to colon, maybe it is getting absorbed because it has not degraded. There may be a degradation, but there the loss may be please remember, in European countries there is a huge detection of diabetics in childhood in children because most of them use raw milk. We boil milk. Okay, basically the bovine insulin is degraded and things. So milk, if you don't boil, has the capability to protect the insulin. If you drink raw milk, there are reports that the raw milk along with the insulin in bovine along with the raw milk of this protein may bypass the stomach and go to colon and start making autoimmune disease. So coming back to his question, what this camel milk is doing, it is protecting the degradation of insulin, allowing the insulin to be in the same formulation, same conformation in the stomach, and that's why it is going to colon and doing the job.
Sir, last but not least, say something about vaccines, whether we are going to get some coronavirus vaccines in near future. What will be its formulation? What should work?
See, remember, don't read newspaper too much. Okay. Remember, you have done on rotavirus vaccine. Things will come call vaccine when it satisfies two things. Number one, when it has neutralizing antibody. Number two, when it gives memory antibody response. Otherwise, it is not called a vaccine. You cannot take vaccine every 3 months, isn't it? You take vaccine once. Smallpox, TB, or TT, you take once. Till now, nobody has shown any memory antibody response. Has anybody shown memory? All these data are coming on first month. Anything you inject, it will come. But they are tremendous results. I am telling you, the results which is coming is unbelievable, wonderful concept. Remember, from a disease to getting human vaccine trial takes 6 years. It took 6 years to develop the rota vaccine. It takes 12 years to make other vaccine. Here, within 1 year, we have discovered the disease, we have sequenced the RNA, DNA, we have made a vaccine formulation, it has gone to clinical trial. That is a tremendous improvement and achievement because of the science. But phase 3 will tell you everything. Because phase 3 will tell you whether it is working on different people, what dose it is working, and how long it is working. Unless it works at least for a year, do you like to take it every 3 months? Can you afford? Can you take it? No.
Yes.
That is the situation. But nevertheless, within the 1 year, in spite of all this pandemic, all these problems, the way vaccine technology has developed, that is really wonderful. Wonderful for all of us, showing that for the first time, any vaccine can be developed within a minimum time. Remember, from the sequence to the first injection, within 3 months. Same thing was happening for rotavirus in 8 years. You sequence, you characterize, you do my study, you evaluate antibody, you stability, then make formulation. 8 years goes, another 4 years goes for trial. So now that the initial 6 to 8 years, we have compressed to 6 months. So hopefully this year end, not this year, next year, mil jayega. Kuch to karega. And remember, till now, for each disease, we have only one vaccine, right? For TT, we have one vaccine. For hepatitis, one vaccine. For measles, we have one vaccine. for the first time, you'll have variety of vaccine. You have a DNA vaccine, you have a RNA vaccine, you have a viral vector vaccine, you have a protein vaccine. And everybody is working. So we don't know who is going to get that. And for the first time, generally we give vaccine to the kids. For first time, you have to give to the older people. Older people have immunosenescence. They don't react. They don't react. Their immune system is weak. Will they be beneficial? So many things are to be asked. But nevertheless, wonderful news from all the company, because the way they have done, the way they have done neutralizing titers. Of course, this is a race. Who will come and who will sell? Those are different things. But the scientific achievement is really, really wonderful. We should all be proud of that.
Thank you, sir. Thank you,
Okay.
sir.
Sorry, Bismita, I am just stopping you. I need to just
Somebody
discuss with sir about this, sir, because as you told that vaccine development takes decades.
Yes.
Yeah, but what is happening here? We are seeing a race. Moderna is coming up, Pfizer is coming up, Sputnik is there, and if we see the Indian pharma companies like Serum and the Bharat Biotech, they are still way far from getting the results here. So what is happening, sir? How it is happening that overnight the technology is so improved that we are getting the results within a year? Even it took I think last week only, it was the anniversary of the COVID, and we are getting the vaccines, and they will
Not
come
last
maybe
week, day
in
before
1.5
yesterday.
years. So
17th November.
is it all commercial interest, or what is that? It looks like more like it's a commercial interest.
Obviously. See, remember how much dose you will sell?
Sir,
Even if
it is
you
millions,
save
it was
10
billions.
rupees for each dose, can you calculate the amount of money?
Exactly.
This is 10 billion rupees here, man. Not a single company is
Oh, sir,
1,000 crore.
more than that. 8 million,
Yes.
8 billions they will be selling.
No.
We just ordered
Let's
1.5
not go
billion,
to
I
that. That
guess.
is not our domain. But the real thing to be happy about that, look how much paper has come in coronavirus. Every month, 400 papers are published. 400. It's difficult to catch with, and they are all published not in low impact factor. They are all in cell, immunity, host, microbe infection, science, nature, Lancet. Every month, 400 papers. How much you will read? Nothing. But no doubt, this time we have shown, from a scientific point of view, unprecedented development in taking the leads to the human beings. Of course, there is a race, no doubt, but please remember the people who are dying. So much people have died. Look at people who are struggling. That is. The other point I want to say about COVID, for these, please remember, this is a higher or a dangerous version of cold virus or coronavirus. See, we don't have a vaccine for cold, right? Every 2 months you get cold. Not like other diseases. We get measles, we get smallpox. Once you recover, we are protected. But we get cold every 3 months, isn't it? That means there is no vaccine. Immunologically, we are not competent to develop a neutralizing antibody titer for any virus which create cold. And this virus, apart from creating cold, it goes inside the lungs. It causes thrombosis, it causes inflammation. I don't know how many of you have read. This virus doesn't stay in the base or the lateral side of the cell. It exactly stay at the basolateral membrane. ये जैसे चूहा छुप के रहता है ना घर के कोने में, वहां तक एंटीबॉडी टाइटर जाएगा नहीं. So it stays there. Now people are coming. Those who have recovered, they are showing symptoms. So the science has to be understood. Please remember, when HIV came in 1984, the similar people who are dealing now, they told we will give HIV vaccine in 2 years. It is 36 years now. We don't know what is the neutralizing antibody titers. Because this HIV, luckily this virus is not mutating. The HIV virus mutates every day. It's like changing the target. You target for this virus, by the time the vaccine is ready, the target is not there. What you are targeting. So these are complex things in science. But the best thing is the way science and our technology and people have been working and competing. But look at the end of the, I wish I could have given you, there are 10 million viral species in the world, 10 million. And out of that, 1 variety is corona. There are 7 corona. 6 doesn't do much. One single tiny of that 1 million virus types is causing that havoc. You have to remember that. And we have been living together with this different virus also. So you have to see in totality. Let's hope something will come for the people, for the old people, for the people who are exposed and who are vulnerable. But
Yes,
only you can hope.
Yes, sir. We will.
Okay.
We are hopeful to get the vaccine very soon. I think we have enough
Yes,
of discussions and enough of maybe emotional. I can say that it's an emotional discussion, definitely. But with this, I would like to thank you, sir. Thank you for your time, the understanding you have given to our audience, our students. I think it's encouraging, and people will be much more interested to take forward this research. Sir, Patra sir, are you there?
Yes, I am here.
Yes, sir.
Thank you, sir. Panda sir, Dr.
Smita,
Panda sir.
one.
Thank you.
Thank you. So nice. So I can log out now.
Sir, yes, sir. I think we will be moving forward with the...
keep the link. I like to listen to in between when I am get little free. Okay.
Yes, sir. Yes, sir. Yes, sir. Sure, sir. Sir,
all the best for the next 2 days.
Yes, sir.
All the best for all of you. Bye.
Thank you, sir. Thank you, sir. Thank you. Thank you so much. Patra sir, over to you, sir. Yes, sir. Please.
So our next speaker is Dr. Veena Menon. Dr. Veena Menon received her BS and MS in Pharmaceutical Sciences from the University of Mumbai, India. Dr. Menon?
Hello? Dr. Ben,
yes.
Call her first.
Yes, I'm here. Sir. Patra sir, I'm here. I think I'm having some problem with my video though. And I'm trying to figure out if I could just, you could continue, sir. I'll just, it's just unfortunate my video is not
she
working.
can actually directly present her things here.
Yes.
So Dr. Veena Menon, and she had her BS and MS in Pharmaceutical Sciences from the University of Mumbai. And then she went on to have her PhD degree from the University of Georgetown, that is in Washington, USA. Her graduate research work in the laboratory of Richard Cladrone involved investigating in the structure function studies of SSK1P, a important regulatory protein of Candida albicans. Then she did her postdoctoral research work at the Beckman Research Institute in California, and then another in the Health Science of Uniform Service University, that is in Bethesda, Maryland. With this experience and some of other experience with HIV and various other viruses, she is currently a faculty scientist at the Department of Clinical Virology at Amrita Institute of Medical Sciences and Hospital. She is actively involved in setting up and expanding the functionalities of clinical virology and surveillance of viral infections with the hospital as well as nearby community. Her laboratory interests including developing newer rapid and novel diagnostics to address the diagnostic challenges of viral and other polymicrobial infections encountering in hospital setting. Apart from all these, the major research focus another is to elucidate that virology and immunological aspects of commonly occurring viral diseases in Kerala particularly, and one of these is flaviviral infections like dengue and chikungunya. She is also a coordinator cum member of the Indian Virus Network, that AGVN Global Virus Network affiliate, that is. So thank you, Dr. Menon, for your willingness to join us, and we are eagerly waiting all the participants to listen your talk. So you please may start your speech.
Sure. Thank you so much, sir. And I should now just start sharing my screen. Is it visible to all?
Yes.
Okay, wonderful. So at the outset, I must I would like to thank the Department of Life Sciences, the National Institute of Technology, Rourkela, Odisha, for giving me this opportunity. I'm truly delighted and honored to be a part of such a distinguished panel of speakers and the distinguished academicians of this institute. Sorry, am I audible? Sorry, should I continue?
Yes, audible.
Yes, yes. Okay. So I follow this talk follows a very wonderful and very informative talk from Sir Panda Sir, and those are really very tough shoes to fill in. And what I am going to present in today's talk here is not so much about the innovative science, but rather something, the nuts and bolts of what it takes once you have an innovation. What does it take to get it out into the market and have it for the consumption of humans, or especially medicinal products. And in a case in point here that I have going to probably talk about is about biosimilars, because I think this is an often term being used nowadays, and there has been a big push from the industry as well as from the FDA to bring the biosimilars into the market. And then what does the what are the regulatory requirements to get these biosimilars into the market? So that's what I will briefly cover, and just it's sort of a brief overview, and I'm not really going to go too much into the details of this. So it's more like an extension of, suppose you have an insulin product that you have innovated in the lab, and then now you want to produce it in big amounts. But having done that, what does it take to bring it out in the market? Let's just get a very brief overview of that. So this is just a brief outline of what the talk is going to probably cover. The most of the slides are going to introduce the subject of biosimilars, then probably stress upon what is the chemistry, the manufacturing, and the controls that is required from a regulatory point of view when you are looking to market a biosimilar. Then what sort of non-clinical evidences for biosimilarity you need to generate to submit for the FDA. And finally, what sort of clinical development is required. So hopefully in the following slides, we will cover these in brief, in brevity. So first thing I would like to address here is what are biologicals and what are biosimilars, and what's the difference between the two? Why aren't biosimilars called biologics? Why are they classified separately? Why are they defined separately? So here's first, to better understand their definition, we also need to know what they are and where they come from. So biologics are first a new class of protein-based therapeutics produced using living organisms. And in this case, it could be the plants, animals, and microorganisms such as yeast and bacteria. They are complex, synthesized using recombinant DNA technology, and they may be either polypeptides or glycoproteins and or nucleic acids. And I know I'm talking to the life science graduates here, and this may be a sort of a no-brainer for you, but just bear with me with this basics. So here is a figure that sort of gives you an idea with respect to the size and complexity of the difference between a small molecule, or what we commonly refer to as generics, versus what is a large molecule and what is a large biological. As you can see in the top panel, if you are looking at the size, if an aspirin is only of 21 atoms, but the human growth hormone is of almost 3,000 atoms. And if you're talking about a monoclonal antibody, it's way more complex than a growth hormone. And you have it is composed of about 25,000 atoms. And concurrently, if you look at the bottom panel, it also sort of reflects the complexity of these molecules. You have, if it's something that's small as aspirin, in terms of complexity, you could correlate it to something like a bike. And if you're looking at a large biological, it will correlate to the complexity associated with a business jet. This is a very simplistic view, and just wanted to point out the size and the complexity differences between a small molecule and a large biologic. And then of course you could further differentiate that into minor complex biological, moderate complex, and truly highly complex. And what is shown here on the scale from left to right is it begins with an antibiotic, and you have the molecular weights, the amino acids, as well as the size fractions of these things versus a monoclonal antibody. And you can see that as we traverse to your right, the complexity and the size of the molecules in study is increasing. So obviously, this is going to influence the type of evidences or the data that you need to generate to get the approval for marketing or for the human use. So with respect to this, where do biosimilars stands? And so usually biosimilars are referred to as similar to the reference biologic. So biosimilar is a medicine that is similar to a biopharmaceutical or a biologic that has already been authorized, or what you call it as in the regulatory parlance as a reference product, and it has already been licensed for a human use. So what is a biosimilar? Is basically a medicine that is similar to something that has already been licensed. So in some FDA or in the Japan and the US, regulatory agencies tend to call this as a follow-on biologic, or in Canada, it is also referred to as a subsequent entry biologic. As you all know, that insulin is off patent now, and so you don't need to always go for the Novo Nordisk insulin product. You have a lot of Indian companies here making the recombinant insulins. Those recombinant products, insulin products, would be referred to as biosimilars. And to get an FDA approval for marketing, they need to satisfy certain requirements. And that is what we will see in the following slides. According to the Indian regulatory agencies, biosimilar is often referred to as a similar biologics. And the active substance of a biosimilar is similar, but not identical to the reference product. Therefore, the regulatory requirements for approval of generics, which is usually a generic of aspirin, where you're talking about a small molecule, are inadequate to demonstrate the quality, efficacy, and safety of biosimilars. So again, to reiterate what they are and what they should not be considered as. So one thing, they are highly similar to already FDA approved a biological product. And most importantly, there is no clinically meaningful difference from the reference, especially with respect to the safety, purity, and potency. Minor differences in the clinically inactive components are allowed. That's not going to create any problems or hiccups in your getting permission to market. And also, it's usually done on the newer version of an existing biological whose patent has expired. But one has to remember that biosimilars are not considered as generics. In other words, what I'm stressing this is because the amount of evidence that you need to furnish to the regulatory agencies is much more compared to a generic molecule. So here is a quick sort of a tabular view of trying to highlight the differences between the three. So you have on the left-hand side, the chemical drugs usually referred to as small molecule generics. You have the biologics, which is the reference compound or the innovator's compound. And then you have the biosimilars. And as you can see here, chemical drugs, they are small molecules. Compared to that, the biologicals and the biosimilars are high molecular weight. They are manufactured organic or by chemical synthesis, while the other two, you have to produce it in a living organism or 6 cells. And that's why you have a lot of inherent and contamination risk associated with that sort of manufacturing process. When you're talking about producing or manufacturing chemicals, they have very few critical process steps. But when you're talking about producing something recombinantly, you all are familiar. There are many critical process steps that needs to be paid attention to. Chemicals, all the available analytical procedures can very well characterize them and ensure reproducibility in its manufacture. While on the other hand, biologics and biosimilars are still, there are a lot of challenges in the manner in which, whether you can truly characterize them. You have the known structure. Many a times you have, with respect to the biologicals and the biosimilars, unknown or undefined structures. And but more importantly, when you're talking about a small molecule generic, they are quite homogeneous in their drug substance, while the drug substance associated with the biological biosimilar is quite heterogeneous mixtures, especially if you're talking about proteins. You can see oftentimes you may have variants being produced as a part of the manufacturing process. And most of the time, these small chemicals are rarely immunogenic, while these biological and biosimilars have a very high potential of being immunogenic in their when administered to the human use. When you talk about the development process, especially with respect to the clinical development, chemical drugs usually have to have, especially with terms of in the generics, usually you just need to produce some phase 1 data. And if you're looking for regulatory approval of already an established a molecule where you're just producing a generic version, you just need to show bioequivalence, and then the automatic substitution is allowed. Unfortunately, that's not the case with biologics and biosimilars. When you're looking at the clinical development of this, you need to do extensive clinical studies, that is anywhere from the phase 1 to the phase 3. And more importantly, you need to have a pharmacovigilance program associated with it. That means even after the molecule gets permission to market, you need to still keep collecting data on the safety and furnish it to the FDA or our Indian regulatory agencies. And the same applies for biosimilars too. And importantly, from a regulatory point of view, you need to show comparability, and there is no automatic substitution in case of biologics. And for the biosimilars, it's slightly different. Basically, what they need to show is just demonstrate a similarity. They need not show that they are better or inferior. They have to just show that they are similar to the reference biology. And obviously, there's no automatic substitution allowed for these molecules, for these products. Now, this sort of figure is just trying to give you an idea as to how usually these developmental pathways occur in the industry, and what the FDA or the regulatory agencies are expecting when they are reviewing a dossier for marketing authorization. As you can see here on your left-hand side is the originator molecule or the biological molecule. And the inverted triangle here is sort of a representation of the quantum of data that you need to furnish to the regulatory agencies. As you can see, you really don't need to generate a lot of analytical or non-clinical data. But as you go into the phase 1 and the phase 3, you really need to generate a lot of evidences of the clinical pharmacology, which basically encompasses the pharmacokinetics and the pharmacodynamic aspects of the molecule. And at the same time, the clinical trial data where you're evaluating the safety, efficacy, and the pharmacovigilance part, including for that particular biologic or the originator molecule. And the green triangle there is just a representation of how small the quantum of data is needed from the regulatory agencies when it is considering a small molecule generic, or when somebody is seeking a permission to market aspirin, because there's already so much information available on aspirin. And if you're going to make it chemically, all you need to show is a lot of analytical data and the bioequivalence in healthy volunteers, and you're good to go as far as the FDA is concerned. When you look at the biosimilars, on the other hand, when you compare it to the originator, you need to show a lot of data on the analytical characterization of your molecule or of your drug substance. You need to show some amount of non-clinical data but if you really have a strong dossier, if you have a good dossier which has very good analytical characterization and very good non-clinical data, then probably the quantum of information that you need to furnish with regards to the clinical pharmacology and the clinical studies is much less. So this is just a sort of a figure that gives you an idea as to the amount of evidences that you need to generate regarding your molecule, especially with respect to the biosimilar, if you're going for an FDA approval. Now, just to give you a brief history of the biosimilars, again, this is a mode that is already existing in the market. The first therapeutic biological was approved in the 1980s, and that happens to be the recombinant human insulin, Humulin, which was approved in the October 1982 by the US FDA. Subsequent to that, I think the first biosimilar, however, happened to be approved in Europe by the EMA, that is the European Medicine Agencies, and it was in 2006. And that was for the molecule somatropin, which is the human growth hormone. And then you have the various brand names there associated with it. Then the first biosimilar monoclonal antibody that was approved by the South Korean FDA was in July 2012. And this happens to be the Remsima from Celltrion, which is a biosimilar of the infliximab monoclonal antibody. And it was approved and marketed in almost 70 countries as of March 28, 2016. The first biosimilar to be approved in US was quite late compared to the EMA. If you remember, the EMA was way ahead in 2006 and granted permission for the marketing. It took a long time for the US to come on board with biosimilars. And finally, the first biosimilar to be approved in US happened only in 2015. This is quite recent, actually. And it happens to be the filgrastim biosimilar. And the FDA just approved the first biosimilar monoclonal antibody. That's again in the US. That's the second biosimilar overall, and which happens to be the Inflectra, and which was approved in 2016. On the same vein, continuing on the same vein, you have the Intas product, actually. It's the Acofil, which is a biosimilar product of the Neopogen, which is filgrastim. Again, it's the first Indian brand biosimilar to be marketed in the EU. You can imagine it really is a good product because it could pass the EMA review process. And this is actually what's Acofil is filgrastim. It's indicated for the treatment of reduction in the duration of neutropenia and the incidence of febrile neutropenia, with mobilizations of the peripheral blood progenitor cells, severe congenital, cyclic, or idiopathic neutropenia, and persistent neutropenia in patients with advanced HIV infection. That's just what's the medicinal use of this product. So why make biosimilars now, and why the big push? So what's the big benefit of biosimilars? It's so you have the originator biological that has gone off patent, and suddenly you have the biosimilars in the scene. So what is it? First of all, biosimilars have a lower development cost. So you can get something into the market in a more cost-effective way. And obviously that means lower cost to the patients. And also because of the low cost, you has improved access to these. You have a larger population can access these medicines. What has benefited its development is you have to have an enhanced CMC package. Now, I would be using this term. This is a very FDA jargon, so to speak. It's called as like I showed before, it's the chemistry, the manufacturing, and the controls. That's the information regarding the manufacturing of that biologic or that biosimilar. So the enhanced CMC package allows for reduced non-clinical and clinical development efforts compared to the originator molecule. And that's why you have a lower development cost associated with biosimilars. And also achieving manufacturing efficiencies using more current methods controls the production cost. And the case in point here being, and what Panda sir showed that earlier, that they have an improved protein expression system and purification system, whereby you can increase the yield of your batches. Surely all that is possible once if you're looking at it from a biosimilar point of view. So this is again another figure that gives you an idea as to what are the timelines with regards to the development of biosimilars and getting it starting from the market for medicinal use. As you can see on the top panel, there is the reference biological development, which takes anywhere between 11 to 12 years for the product to come out into the market. While if you are looking to develop a biosimilar, it is usually done within 8 years maximum. There's a 7 to 8 years. That's all. And the total cost also you have is far less compared to what it takes to develop an originator molecule. And so what is good as of now, why you think it's relevant for us to know about biosimilar, is that a lot of these biotherapeutics are now coming off patent, and which means it is opening the doors to biosimilars. So you have a lot of biotech companies in India that's going big time into production of biosimilars. So there's a big boost for the academia, the students. We really need to have trained students that can take on this mantle. Now let's look at the global regulatory landscape with regards to biosimilar. Again, the regulatory guidelines vary a lot and globally. And the criterias of variation usually either temporally, that means the time point at which these guidelines were issued, and also in terms of content as to what are the differences. Like Indian regulatory authorities would have some minor changes when compared to what is required for the US and the European markets. So definitely there are differences in the guidelines. And what you see here, bottom figure here is just the evolution of the biosimilar regulations. That this is the evolution of the regulatory scene with regards to the biosimilars, and the time period that is spanning this figure here is from 2004 to 2014. So it's not really a very updated figure here, unfortunately. I must tell you that although in India we have we were first introduced the regulatory guidances in 2012. We have revised this, and now you have the latest version is the 2016 versions, and it's quite in tandem with what is there in the US and the EMA. So we definitely have very high stringent guidelines and good standards set for the industry to help the industry or to guide the industry in coming up with a good quality product. So what are the intent of these regulatory guidances? First, some themes are consistent. So what it means is that what it helps is that there is a stepwise assessment of similarity beginning with a strong analytical or CMC package. So your CMC package, when you are generating evidences and going to furnish it to the FDA, that dossier should have very strong analytical data that shows that your molecule is highly similar to the existing reference biologic already there in the market. And this assessment is usually done in a stepwise manner. And the one area that is less consistent is the need for non-clinical in vivo studies. So this is something that that the FDA or the regulatory agents are still quite ambiguous about. You could have a conversation with them if you are an industry partner and you want to market something and you're not quite sure if you have a very good CMC package, do you still need to produce a lot of animal studies? And this is something that the FDA on a case-by-case basis would consider. It all depends upon your quality of your analytical data. So the EMA, the European Medicine Agencies, appears least likely to request in vivo studies if the analytical package supports similarity. And obviously the US FDA appears to be getting closer to what the EMA expectations are. And with regards to the other countries, some, especially in India, we still require to furnish some animal studies and at least one in vivo toxicity study, even when the clinical data available for the proposed biosimilar is there. So, which means you need to generate some animal studies, especially toxicity studies with regards to the approval of biosimilars in India. So what is needed to show biosimilarity? What sort of analytical package, what sort of non-clinical and clinical data you need to furnish? So the goal of a biosimilar development is to demonstrate no clinical meaningful difference from the originator based upon the totality of the evidence. So it's not one single assay or one single method that will be enough to show biosimilarity. You have to furnish a lot of evidences, and the evidences in total, that is the overall outlook of that evidence, should demonstrate biosimilarity, meaning that there is no clinically meaningful difference between the originator molecule. So just want to reiterate, not to re-establish the safety, efficacy, and overall benefit. This has already been shown by the reference biologic. So basically you have to show that this is very similar to the reference. That's all. So what does this usually entail? It entails some combination of analytical, animal, and or human testing. Again, the testing is not going to be safety, efficacy, and overall benefit. The safety will be again, the clinical data would again be to show the biosimilarities similar to the reference biological. So the analytical testing is usually more extensive versus what the originator did. And the animal and the clinical testing is less extensive. And the testing, as I've been saying repeatedly, the testing is often comparative. So currently there is an effort to minimize or eliminate animal testing even more when scientifically justified. So let us now look at what is the chemistry, manufacturing, and controls associated with biologics and obviously with biosimilars. So what you have here is just a wonderful figure, I should say, that sort of explains to you the quantum, the amount of evidences that you need to generate in the preclinical phase as well as in the clinical phase in that developmental pathway of biosimilars. As you can see, the amount of CMC data that you need to furnish in the preclinical phase is much higher, while in the clinical phase you have the novel biological development program will have much more clinical data that needs to be furnished. So here you have the pink or whatever you call brown part representing the originator molecule, novel molecule, while you have here the red is the biosimilar. So obviously you need to generate a lot of CMC data, which usually includes the analytical, that is the physiochemical, structural, and the bioanalytical data, along with the in vitro functional or the binding data. You need to generate some in vivo functional PK/PD as well as toxicological data. All this is just to minimize the downstream risk of failure. So the first thing when you're looking at CMC is to understand the mechanism of action of your reference biologic or the biologic that you are trying to produce. So understanding the mechanism of the product enables you to understand the clinical relevance of any observed structural differences. And this in turn enables us to design an initial control strategy to monitor and control for any risk associated with deviations. So there are certain amount of deviations that are allowed as long as it has it is not reflected in the clinical effect or the biological effect. Those are tolerated. There are specifications set for allowed impurities. So to understand if a certain change is going to influence your development pathway, you need to really understand what is the mechanism of action of that product. Say, for example, insulin. It is going to bind, the way it is elicits its action, it binds to the receptor, insulin receptor. So if you have any mutation there that is going to affect the binding to the receptor, obviously that's not going to really be a correct procedure or manufacturing procedure. You will have to take care where you are reducing the risk associated with those mutations. So what does that mean? When you look at it from the analytical point of view, you need to generate the evidences to confirm that the amino acid sequence is identical. Then you have to assess and show data of any post-translational modification, including glycosylations. Then you have to show data regarding the product degradations, denaturation or aggregation or oxidation. When you're doing HPLC for purification, you have to show that there are no dimers being formed. There are, those are all below the limits, etc. Product-related impurities should be identified and they should be sequenced and characterized and shown that it has no effect on the safety and the efficacy of the product. And process-related impurities, if you're growing it in a eukaryotic cell system, or if you're growing it in a bacterial cell system, you have to have limits of production of endotoxin or the host cell protein. You have to have limits. You should not have any adventitious agents entering into your final product. So again, I mean, this is, you all are very familiar with this, but just for the sake of this talk, I just put it up here so that I can segue into whatever I wanted to say in a much streamlined manner. So what we have here is production of recombinant proteins. Again, you could either use a unicellular system or a multicellular system. You have currently in the market bacterial, yeast, mammalian, insect-based unicellular system. You could also, now you also hear of transgenic plants and transgenic animals being used as a system to produce recombinant proteins. Again, here is generally how does this happen. This is a very basic figure, and really, I really don't need to explain this. You have the gene of interest, which is cloned into a vector, and it is then transformed into a bacteria or a cell or a mammalian cell, and you have a genetically modified organism expressing the protein of interest. So what happens once you have identified your clone and then you have, put it into the storage? Now you're going to use it to manufacture. So what does the manufacture biological usually takes? Again, this is very proprietary, so that you really don't have the details of how insulin is prepared by maybe Sanofi, and how it is prepared by, say, another Indian company. So it's a very proprietary information. But generically speaking, what you see is that initially you will have something where you develop and establish a master cell line bank. Subsequent to that, there is a subculturing process where the cells are expanded for isolation and purification. Sort of the fermentation process is happening, and then you purify it. So there is an upstream development process and there is a downstream development process. Subsequent to that, you have your drug substance in hand. Then what you do is you do a lot of in vitro characterization and you do the stability. You will formulate it into a product, and then you will have to do time-bound stability studies to show that you still have the active substance right there, not losing its function. And then finally, having established all the stability requirements, you will finally formulate, fill, finish, and package. And more importantly, subsequent to approval, that although you might get approval with your clinical trial batches, you are constantly monitored by the regulatory agencies, especially the biomanufacturing process, just to ensure the pharmaceutical quality. So it doesn't mean that the story ends with approval. You're continuously being monitored by the FDA to ensure that every batch to batch, you are really getting a quality product out into the market. So how does this happen? The FDA has a lot of guidance documents to help the industry, guide them through the whole process of manufacturing and marketing a biosimilar. And I have listed here ICH documents. What's the ICH actually? Actually, ICH is the International Council for Harmonization of Technical Requirements for Pharmaceuticals for Human Use. So each of these sections here, that is that Q5A, Q5B, Q5C, as you can see, these are the sections in the ICH that will give you information as to how to ensure viral safety. What sort of evidences do you need to generate? What sort of documentation you need to show to the FDA to prove that the product is free of viruses, genetic stability, product stability. I wouldn't go into all the details, but this is just there. Anybody who's interested can always look this up. These are very well characterized data there that the industry needs to follow to come out with a good product. So just giving you an overview, a flow chart of what is the process usually in the industry and what sort of regulations are applied at each of these stages. As you can see in the genetic development, you need to have really good, you have to fulfill all the requirements of the FDA associated with the Q5A, Q5B, 5D, and the 5E, especially when it also goes into the generating the master cell bank and the working cell bank. And then associated with the actual bioprocessing, that is the biomanufacturing bioprocessing, which includes the culture, the fermentation, the purification, and finally you have the drug substance. These are governed by the Q5A, C, 5E, 6B, and so on and so forth. Finally, you have the M4, which actually here, as you can see, M4 and the M2 is the, this is called as the complete technical document or the electronic complete technical document for that particular drug product that you are going to have to fill it out for submission to the FDA or the regulatory agency for marketing approval. So I'll just very briefly, and I know this might get a little tedious and monotonous, so I'm not going to really cover all these steps, but I just wanted to give you an idea what it usually means. What are these Q5C and Q5D? What are the sort of evidences they say they would like to see? So if you're talking with respect to the cell culture and the harvest, the sort of information that you should have is you should have a very elucidate a good flow diagram. That means from the working cell bank up to the last harvesting operation, you should include all steps, whatever the unit operations are, and any intermediates that are produced in the flow chart. All relevant information for each stage, the volumes, the population doubling time, volumes, times, etc. Then what are the critical steps and the critical intermediate, along with the specifications for each of these steps. In addition to that, usually these guidelines also required for you to give a description of each process steps, which would include scale-ups, culture media, other additives you have added, the major equipments you have used, what sort of process controls you have included, what sort of in-process tests you have done, what are the operational parameters, what is the process steps, equipments, intermediate, everything along with the acceptance criteria for these. Then information on the procedures used to transfer material between the steps, the area, the equipment, the GMP requirements, everything needs to be documented in your dossier. Then whatever, what are the criteria that you have come up with in your biomanufacturing for rejection of the harvest and the premature termination of the culture, and what is the single harvest production and what is the multi-harvest production details too needs to be furnished. When with respect to the purification or the downstream processing also has a very similar sort of requirements. You should have a flow diagram from the crude harvest to the final filling of the drug substance. All steps and intermediates and all the relevant information for each stage should be well documented. You have to capture all the details associated with any of those steps, and identify the critical steps, and then set specifications and limits for any deviations for in those steps. So you should have very good rejection criteria also included into your quality management system when you're talking about producing these in really large scales. And of course, a description of each of these processes, which is quite similar to what we saw for the upstream. So I'm just going to gloss over it. And finally, you will come to the filling, storage, and the transportation. You should, again, even for these steps, you should have a complete description of the filling procedure and the process controls associated with filling procedures, and whatever in-process tests are there, what are the operational parameters associated with it, and what is the acceptance criteria that is that should be provided for. Then the container closure systems used for storage of the drug substance, the storage and the shipping conditions, everything should be documented in detail for the FDA to review. So, so these are some of the key points with regards to the biomanufacturing. Now, when you are looking at the other important aspect, especially when you're looking at producing these biologics or biosimilars, we all know we do it in a living system. So there are a lot of inherent risks associated with it, especially with regards to the purity. And if you are using a cell line, you take or a mammalian cell line to produce your recombinant protein, then there are a lot of known and unknown viral contaminants that might come out in your drug substance during the purification process. So you have to make sure that you are reducing the risk associated with any known and unknown viral contaminants. So what are usually the raw materials and the products that can, where this sort of known and unknown viruses play a huge risk or pose a huge risk, is the cell lines itself or the raw materials that they use. Suppose you're using a enzyme. Most of these enzymes are produced in turn are produced recombinantly, either in a bacterial system. So maybe when you are doing on the bench work, you really don't need to have a very highly pure peptidase enzyme, or for that matter any other restriction nuclease enzyme. But if you are going to use it in a process where the final product is going to be used for in human use, then you have to have very detailed specifications and quality control done on these products. So the product needs to be tested at appropriate stages. At the same time, you have to make sure that your the processes that you're using, the fermentation process, the purification process, those processes have the capacity to clear the infectious virus. So what are some of the things with relation to viral safety? You have the cell lines and the raw materials. You have your master cell bank. So you have to keep checking it for the presence of any latent or persistent virus infections, endogenous, retrovirus, etc. Those are some of the elements that you need to characterize and inform the FDA of whatever data you have. And you also have to make sure that introduction of adventitious virus during production is kept at the minimum. Especially don't use contaminated biological reagents and excipients. With respect to product testing at appropriate stages, you have all these cell lines need to be tested at regular intervals for retroviruses, endogenous viruses, infectivity, electromicroscopy should be done, RT-PCR data should be provided, specific tests, etc. can be done on these. At the same time, you can also do non-endogenous viruses. You can do some in vitro tests, in vivo, and show that there is no antibody production, etc. for these. And it can also be done for the unprocessed bulk. Again, other than viral safety, you also have to show the genetic stability of the platform in which you are expressing your biosimilar. So you have to make sure there are no potential mutations in the expression system. Obviously, you need to do elaborate nucleic acid analysis and protein analysis. You have to make sure that you want in your clones that you have generated, there are no mutations are happening with every division it undergoes. When you're deriving the cell substrate, so for example, your insulin, you have to have very good information collected with regards to the origin, source, and history of the cell substrate. So a lot of elaborate literature search should be done, and R&D development information should be provided. Published data, historical data from other sources, etc., should be included in your dossier. Then characteristics of the cell substrate, the species, the strain, the genotype, the phenotype, characteristics, the generation level, any toxin production. If you're using E. coli, what sort of strain are you using? If it must be a mutated strain, you have to make sure there is no toxin production associated with that E. coli host strain. Any sort of biohazard that is associated with your expression system should be identified and documented. In addition, there's a biological purity should be ensured. Exposure to infectious agents should also be identified. Any exposure to infectious agents should be identified and documented and reduced. Actually, if it is going to pose a risk, it has to be eliminated. Generation of the cell substrates, again, all the procedures that are used to obtain the cell substrate, whether it is transfection or transformation, what sort of vector system did you use, all that should be well documented. Then it is required that once you have identified your clone, you have to generate a banking system. And how to have this banking system is also using the regulatory requirements. It is not something that we do in our lab. It is something that you have to have a standardized format in which this is done. And again, you have the regulatory agencies giving you the guidance as to how this needs to be done. So your cell banking system should be two-tiered. You should have a master cell bank, one that is directly derived from the initial clone. And then you will have something called as a working cell bank, which is derived from the one or more vials of the MCV. And all this should be appropriately characterized. That means you will have to take one or two clones from your working cell bank as well as your master cell bank. You produce a product. You have to do a lot of analytical characterization and show that there is no batch-to-batch variations. So all this also, when you are banking, all the procedures needs to be well documented. There should not be any ambiguity in the procedures that you are used for banking. And there should be complete cell bank characterization. That means at regular time points, you will have to do the identity and the purity check for these, provide data to the FDA for regarding the purity and the identity. You have to show that there is no sequence change, that there is no batch-to-batch variations, and then there is no adventitious contaminants that might influence your final product. Again, your cell substrate stability. There are two concerns associated with the stability part. You have to make sure that your production is consistent and retention of the production capacity during storage under defined conditions. It should not be that while you're producing, you have the final product, you have, say for example, you're producing insulin. We know that the dimer levels in the insulin product, the final product that is going to go for human use, should have very low amounts of dimers or any other charge variants. You have to make sure that even after storage in 2 to 4 degrees, that these dimer levels are much low. So in other words, you have to show that the stability of the product is maintained during the storage under defined conditions. And stability during cultivation for production, at least 2 time points should be examined. One using cells that have received a minimal number of subcultivations, and another using cells at or beyond the limit of the in vitro cell age for production, or they're called as a LIVCA. Then evaluation of cell substrate here, again, consistency of the coding sequence for production is for production use or beyond. You can do this by doing the nucleic acid testing or the product analysis. In addition to that, specific traits can also be evaluated, which would include morphological characteristics, growth characteristics, etc. You have to just show that the growth characteristics of a E. coli has not changed with storage. Now, what about the banked cell stability under defined storage conditions, usually generated during production of clinical trial material? You have to make sure the cell is still viable after reconstitution. Then you should have a method for monitoring of the banked cell stability. And if the viability of the cell substrate is not significantly decreased, generally no further testing of the MCB or the WBCB is considered necessary. So those are the requirements when you're looking at from the CMC point of view. Now let's look at what sort of clinical and non-clinical data you need to generate for biosimilars, especially what sort of comparative data you need. So the EMA and the US FDA requirements for biosimilar development is almost similar here. What you have on the left-hand side is the originator biological product, where your step one, you're generating a lot of quality data, which usually includes the physical, chemical, and the biological studies. Then you have the non-clinical data, which is usually a lot of animal studies or the preclinical studies. And then subsequent to that, you have the clinical data. When you're looking at this with reference to the originator molecule, the biosimilar, as you can see in the diagram shown on to your right hand side, most of it is just called as comparability data. So you really have to always show a head-to-head comparison. If you're doing any analytical assay, you have to do it as a comparative analytical assay, and you have to show that in all respects, whether it is the physicochemical, the biological, the non-clinical, or the clinical, your molecule, the insulin that you produced in your manufacturing facility is very close to the reference biology. Or maybe you have picked up Sanofi's product, Sanofi's glargine you have picked up as your reference biology. Then you have to show that Sanofi's glargine and your glargine is very similar. Again, objectives, again, this might be a little redundant now, but again, the design and development requires an in-depth understanding of the structure and function of the reference, and basically to establish a target quality profile that can be used to evaluate any potential analytical difference and the relationship to function. It is uniquely important to define the critical quality attributes. This is a very important term that is often used within the FDA. That is the critical quality attributes. What are those? Those that impact the pharmacokinetics, safety, and efficacy of a proposed biosimilar via thorough analytical characterization. So if you have thoroughly characterized your product, especially with reference to the reference biologic, then a lot of the times you don't have to, you can do away with the animal studies. So what are CQAs? They are defined as physical or biological properties of a product that should be characterized and controlled within an appropriate range to ensure product quality. Like for example, with the insulins, you have to make sure that the dimer content is much below the range that is needed for a product to be safe and not immunogenic. Similarly, what the amount of host cell proteins that are there? You have to make sure what are the critical quality attributes is something that is set up from product to product. It will change from product to product. What is important for insulin may not be important for a growth hormone. So again, it may vary, and it will depend, a lot of it will depend on the biosimilar considered for the manufacturing. Any manufacturing or synthesis differences, if there is, you have to make sure that the product and the process are important attributes to maintain similarity. That means product itself in this case could be, say, infliximab, any monoclonal antibodies, and the process, that is the process by which you're going to produce the monoclonal antibody. Both are important attributes to maintain biosimilarity. So the process optimization towards similarity and precise control during manufacturing should be there to maintain similarity. And that is how you ensure the quality of the biosimilars. So how is that usually done in the industry? They do something called as a QBD. That is the use of a quality by design strategies that allows a proposed biosimilar to achieve highly similar, especially high similarity of the complex features associated with their product, thereby ensuring quality and safety. So variations in the source of raw materials, the type of equipments, any changes in the operating process parameters, manufacturing facilities, all can influence the product quality. And so the critical structure activity characteristics, especially of the protein therapeutics, include primary, secondary, tertiary, and higher order structure, as well as the post-translation modifications. So if you show analytically, if you show that that although you have sourced a different raw material, different type of equipments, and different type of operating processes, but essentially you're getting the same insulin molecule out, which is very similar to the Sanofi's molecule, then that is fair enough. That is good enough for the FDA to consider it for approval. Then the importance of host cell used. Again, just wanted to give you an example here. You can see that the importance of the host cell or the expression system that you're going to use for these biosimilars. As you can see, 3 different batches of a biosimilar product in the same cell type. Okay, and you can clearly see there is so much batch to batch variations when you are looking at the ADCC activity parameter of that reference product. This must be probably a monoclonal antibody. And you can see that it is really not the same. There is big significant differences from batch to batch. While on the other hand, you have a biosimilar that is really showing when you are looking at a specific biological activity as person-specific analysis, you have 2 products here, again produced in a totally different cell system, a bacterial system, the other one is a eukaryotic system. And you can see it's very biosimilar. So this is good to go. The FDA would love such data, while the other one is most likely to be rejected. So one has to be very careful about all such things. I mean, all these are the small details in which the FDA will really raise a red flag. So use of the same parietal cell line as a reference product does not guarantee that this critical quality attributes are met, will be similar in the proposed biosimilar, and that different host cell systems can potentially still have similar functional attributes. Now, what sort of non-clinical studies are required? So that covers the CMC portion. Now let's just look at what are the non-clinical studies. Again, these should complement the structural analysis, especially impact of the structural differences on activity. Also used for the selection of the drug candidates, product release, and stability assessment. And it can predict type and extent of subsequent in vivo and clinical study needed to establish biosimilarity. So the sort of non-clinical data, that is the in vitro, the bioassays, if you really get good data, then the FDA will really consider if you really need a full-fledged clinical trial. So the role of non-clinical studies in biosimilar comparisons. Again, this is a risk-based stepwise approach, and it favors a preclinical program that prioritizes the use of in vitro studies and includes in vivo studies in relevant species as the second step. So if you really have robust in vitro studies, which really convinces the FDA that the product is good and extremely comparable to the reference biological, then like I said before, in vivo studies will become in a relevant species would be not the priority. Only if any additional information can be obtained, only then will it recommend to go in for an animal study. So also that, one of the drawbacks is there is very low sensitivity of animal testing, animal studies, and sometimes they are not suited to detect subtle differences between the biosimilars and the reference products. And so going for animal studies may not be really helpful in that manner. So in that case, it's something that the FDA will consider to waive off. Also, in some cases, when unforeseen differences emerge, in vivo evaluation may be warranted, and only if the relevant animal models are available. With respect to the in vitro and the bioassays compatibility evidence, so no specific details are available with the FDA regarding the structural, functional, preclinical, clinical studies, evidences of biosimilarity. The biosimilar developers have to develop their own plan, and they have to do that in discussion to the FDA and sort of convince them that the data is sort of ensures biosimilarity. The nature of comparability studies vary depending on the product type, and it is determined on a case-by-case basis. Qualitative and quantitative differences are demonstrated, and they should not have any or no impact on the clinical performance of the biosimilar related to the reference biology. Again, clinical PK/PD studies as well as efficacy and safety data are always required to confirm that there is no clinically meaningful differences. Now, I must say very recently, the FDA, US FDA and the EMA are moving away from this requirement, and they are saying that if you have, because technology has brought many high throughput investigations and analytical characterizations possible nowadays. And the discussion now is that, you know, the FDA may consider waiving off even clinical trials if you have a good CMC package associated with your biosimilar. Again, in some other in vitro bioassays like receptor binding assays or apoptosis or cell collection assays, these are all included at the parts of your docket or dossier. You can have the ADCs, the ADCVI, the ELISA. Toxicological studies would be either repeat dose toxicokinetic studies, measurements of titers, cross-reactivity, neutralization titers. And however, what is not needed with respect to the biosimilar is the safety pharmacology, the reproduction toxicology, mutagenicity, as well as the carcinogenicity studies. We don't need to produce these because the reference biological has already produced this information. And so there is no need for reinventing the wheel with respect to the biosimilar. Again, if you're looking at a biosimilar, a monoclonal antibody as a biosimilar, you have some of the physical chemical property listed on the left-hand side and the biological properties that are listed on the right-hand side. And you have all these sort of assays that can be done and data that can be generated to characterize this monoclonal antibody. These are the things. So your binding assays could be either the biolayer interferometry, flow cytometry, SPR. And if you're looking at cellular assays, you can generate neutralization assay data, ADCC, as well as the CDC data, dependent activation data. Design considerations for what about the in vivo studies? Now that you have shown enough of in vitro bioassay and analytical data, let's look at some of the in vivo studies that is usually requested. Again, the exact purpose of the study should be carefully considered, especially when you're looking at the number of animals that needs to be included in the study. You have to keep it to the minimum. Mice or rats, rather than monkeys, are often suitable to assess the PK properties of the monoclonal antibodies. So even when there is no pharmacological activity present in the rodents, they work very well as a animal model to show PK comparability. And testing in one sex should be considered wherever possible, but in some regions and some regulatory agencies insist on ensuring that there is a balanced sex ratio between the animals included in the biocomparability study. And assessment of recovery generally is not necessary with respect to the animal studies. Additionally, testing at one dose is usually sufficient. Dose should match one of those used by the originator and should not saturate dose response so that differences between the proposed biosimilar originator can be more readily detected. Because you have to finally realize that the exercise of the animal study is to show comparability and not uniqueness or better or inferiority when compared to the reference biology. Again, relevant control for the proposed biosimilar is the originator's material. And so you cannot use a vehicle control group, and it's usually not needed. And testing of a single reference product is sufficient. Where there is a regulatory request for at least a different version of the reference product, this can be achieved by the CMC characterization and a clinical PK/PD bridging study. That should be fair enough. You really don't need to go into a animal study to show the same. Again, with respect to the PK data, if the outcomes of the in vitro comparative studies raises concern, then perform the following single dose PK studies. If for some reason your in vitro comparative studies are really not very robust, in those cases, the FDA may consider single dose PK study to further evaluate your product. Preferred animal are usually non-human primates, NHPs, especially if the reference product used primates. And you have here, there was no GMP analytical difference between the 5 batches. You can see how the batch 2 and 5 performed when you're looking at the PK pharmacokinetics of the product, while the batch 1, 3, and 4 worked very well, but batches 2 and 5 failed. And you have to mind it, there was no analytical differences between the 5 batches. So yes, sometimes you require the PK data to capture differences. Perform a single dose PK study in mice if it is bioactive in rodents, not required but valuable. And the bioanalytical assay does not have to be validated for this study. So you're good enough. It doesn't have to be a validated method. Then what about the in vivo toxicokinetics and the immunogenicity studies? Well, it's an essential. Establish in vivo comparability of the toxicokinetics and immunogenicity using a relevant animal species, generally primates. You dose the reference product as well as the biosimilar and use it as in a head-to-head comparison. You can use a single repeat dose, 4 dosing cycles per study, or as per clinical dosing regimen. If you're looking at a product which has a half-life of about 2 to 4 days, then weekly dosing cycle. And if the half-life is 2 to 3 weeks, then monthly dosing cycle. Single dose level, 1 gender, no recovery group may be acceptable. If it is not, then you have to use the minimum dose levels. Which are the minimum dose levels you have to choose? You have to choose a high dose, a low dose, and the control. And then usually you have 3 animals per sex per dose level for necropsy, 2 animals per sex for the recovery group. If the reference product did not have a recovery group, then the biosimilar does not need to show a recovery group. What all needs to be done under this study? You perform the standard battery of health assessment testing, perform the toxicokinetic measurements after the first dose and the last dose. You perform the immunogenicity testing. You look at the levels of antibodies generated, and also other non-clinical studies at safety pharmacology that are usually not required, which sort of a little repetitive in this slide. So that takes care of the preclinical data or the animal data that is required for biosimilarity. Now, what about the clinical studies, the action in healthy volunteers and in the patients? Under the stepwise comparability exercise, the clinical program should be designed to match or commensurate with the level of evidence obtained from the preceding steps that support comparability. So the sort of CMC package, the sort of non-clinical data you generate will inform your the design of the clinical study. Again, clinical studies will include phase 1 and phase 2 and 3. Phase 1 would be taking care of the PK/PD, safety and immunogenicity, while the phase 2-3 usually looks at the safety, efficacy and immunogenicity. And again, this is always done in comparison. It's not a standalone study. Regulatory FDA, the US FDA and the EMA requirements on clinical comparisons is head-to-head comparison of PK and PD with reference biological in humans. At least one adequately powered randomized parallel group comparative clinical trial, preferably double-blinded, to compare the efficacy and the safety of the biosimilar with the licensed reference product. Demonstration of equivalent, that is, it should neither be low nor higher, because again, if you have higher, it's also a problem. So it's not, you should not be showing superiority either. Efficacy is needed to adopt the dose recommendations established for the reference product. Important that the trial enrolls in a homogeneous patient population in a clinical condition that is most sensitive to detect the relevant differences in the molecule, if any. So finally, say for example, you're looking at insulin, then it is important that in the final safety and efficacy study, you are including diabetic patients and that you're not really including only healthy volunteers. Again, the pharmacokinetic similarity needs to be established for assessing the product-specific exposure, that is serum concentrations across time, absorption, distribution, metabolism, and elimination. That means it should include all the ADME requirements. The design elements, what are usually incorporated in the PKPD, either it could be single dose, steady state, repetitive measures, crossover. All of these design elements need to be justified against what is known about the reference product. The pharmacodynamic studies can be performed if suitable markers are available. In some cases, PK parameters like area under the effective concentration may serve as a surrogate for the pharmacodynamic comparability data. The PD studies, usually they generally expect PK studies to be conducted as well as the PD studies, especially if a suitable marker is available that gives a measurement of the pharmacodynamic property of the molecule. Measurement of at least one biomarker, a relevant surrogate to the dynamic effect of the drug, must be conducted in a subject population most sensitive to any differences in the dynamic effect. So you have to actually, when you're looking at the PD studies, at least one biomarker needs to be used, usually performed in the targeted patient population at a dose in the steep part of the dose response curve. So for example, you have the erythropoietin where there is a good PK-PD data available, since hemoglobin level is a reliable and easily measurable PD marker which correlates well to PK. If you're looking at insulin, then you can always look at the A1C levels, which is a very good PD marker for making sure that your glucose levels are being controlled well by your biosimilar insulin. For that matter, filgrastim, which is a granulocyte colony stimulating factor to the reference product. Equivalence could be demonstrated for the serum concentration levels by measuring the absolute neutrophil count, and even more importantly for the CD34 positive cell count, which is a marker for the ability of the granulocyte colony stimulating factor to mobilize stem cells. So those two can be your PD markers for their clinical trials. The margins defining what is considered clinically comparable must be defined a priority. So obviously that's a no-brainer there. Again, in addition to the safety and the efficacy, immunogenicity has to be also compared. We all know biologics are large molecules, often referred are more capable of triggering a anti-drug antibody response. And the ADAs may recognize other epitopes favoring the formation of immune complexes that may eventually affect your PK/PD profile. And the immunogenicity of your biologics can be both product or process related. You may have some advantageous material coming in from your manufacturing process, ends up in your final product that may turn out to be immunogenic in the human use. And so you have to actually show data that that the immunogenicity is also comparable between the two, reference and the biosimilar. And this should continue through the clinical trials also. Efficacy evaluation and comparison, again, this has to compare, like this is being a little repetitive here, I'm sorry about that, to confirm that the biosimilar has neither decreased nor increased efficacy compared to the reference biologic. The efficacy endpoint can be that of clinical benefit or alternatively a meaningful surrogate for efficacy. A concept introduced by FDA called as indication extrapolation. What does it mean? Through which one sensitive indication is studied in the phase 3 efficacy trial. If the data is sufficient, it is extrapolated and the product is approved for all the same indications as a reference product. So maybe you have the reference product being used in multiple indications. And then when you're looking at the clinical trial data, say for example, you're looking at filgrastim. Filgrastim can be used in various conditions, post-radiation or maybe in a highly immunocompromised or post-transplantation. You don't have to have clinical trials looking at each of these indications. You can just show biosimilarity to your reference biological for one indication. Maybe you enroll all transplant patients, post-transplant patients into the study, and they have done very well. They have the ANC reconstituted very well, meeting the reference biological data. Then obviously the FDA will approve your product for the other indications for which the reference biologic has already got the permission for. Again, the study design may include a single switch or transition phase in which the study population, the comparator arm, is re-randomized to either receive the proposed biosimilar or continue in the comparator arm. Again, these are all design variations of your clinical trials that the biosimilars can undertake. Again, this again differs from a case-to-case basis, and you can have discussions with the FDA and to finalize on the design that will best provide data to the FDA so that it can review the product. Again, this should ensure there is no immunogenicity concerns after switching from the reference product to the proposed biosimilar. Safety concern for biosimilars, safety is assessed in the same study as efficacy. The choice of patient population should also consider sensitivity for detection of differences with respect to safety. Usually this may be a population for which the investigational product is used as a monotherapy. So finally, I just wanted to conclude. So that takes care of the clinical data. hopefully, this has not become too didactic. So I just very quickly try to recap on what was talked about in the earlier slides. Again, if you're looking at the biosimilars, you can see that you have a lot of the timelines of your development program for biosimilars. You have the CMC, the preclinical section, all starting off initially, where you have the structural and functional testing. You have the animal studies. Subsequent to that, you have the phase 1 and the phase 3, where it covers the PK/PD, immunogenicity, efficacy, and similarity. But you should significantly note here that the CMC continues to develop as you are doing these clinical studies. It doesn't stop. As you can see, the structural and functional testing, there is continuous testing of multiple batches, stability studies are ongoing, and batch release testing are being done. And of course, when you're looking at the clinical part, you have to make sure all the regulatory strategies are being met. And finally, the bottom panel actually doesn't have relevance in our society because we are not governed by these CMAs. This is a slide that I borrowed from the US FDA thing. So this is something that how these sort of biosimilars play a role into the policy, especially when you're looking at the health insurance policy. So that's not relevant to our society, but just wanted to put it out there. And you have to, one thing that needs to be highlighted here is that although you have got marketing approval and the product is ready to go, you still need to continue with your pharmacovigilance and patient reported outcomes associated with your product. So you continuously need to furnish that data to the FDA. So with this, I like to really thank you for all your patient hearing. And that's it. Thank you, sir. And thank you, Bismita.
Yeah, so,
thank you, Dr. Menon, for your talk. It's really, a splendid job. And students will be benefited, and those who are rigorously working in this field, they will be benefited much. So thank you once again. And I think the forum is now open for discussion of your topic, and I request you to address the gathering. Thank you.
Thank you, sir. Thank you so much. Thank you, Dr. Veena. It was indeed a very nice, informative, loaded with so much information that definitely our students going to benefit. Mostly it is going to benefit the people who are really looking forward to translate their findings, their patents, and their work into some translational research and get some product out of it. So they would understand what are the steps and what are the things needed to be done, how to take up the follow-ups, and what are the steps, critical steps involved in each aspects. So before that, I think we have some questions from YouTube, and there is a question. I think we can take on questions, at least 2 to 3 questions, right, Dr. Veena? Hello? 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 Dr. Veena, are you there? Hello? Hello? I think we can take on the questions afterwards with Veena ma'am, and probably will circulate all of the people who has asked questions, and it will be circulated. So with this, I don't see Veena ma'am with some network problem or something. She's not here, I guess. So with this, we'll conclude this session, and we'll again meet up according to our schedule. We'll again meet up at 2:30 for the next session. So for now, we are saying bye to the session, and we'll again meet at 2:30 p.m. For the next session. So we have 3 speakers in the next session, and we'll see you there. Okay, thank you all. Thank you for your participation. So we look forward to meeting again at 2:30. Thank you all. So with this, I am ending this session. Okay.